Experiment 12. Same experiment as the two last, and here again the leaf at the end, enclosing the cheese opened before the page 304 opposite end, with the albumin, but no further observations were made. Experiment 13. A globule of chemically prepared casein, about 110 of an inch in diameter, was placed on a leaf, which spontaneously opened after 8 days. The casein now consisted of a soft, sticky mass, very little, if at all, reduced in size, but bathed in acid secretion. These experiments are sufficient to show that the secretion from the glands of Dionea dissolves albumin gelatine, and meat, if two large pieces are not given globules of fat and fibroelastic tissue, are not digested. The secretion, with its dissolved matter, if not in excess, is subsequently absorbed. On the other hand, although chemically prepared casein and cheese, as in the case of Drosera, excite much acid secretion, owing, I presume, to the absorption of some included albuminous matter, these substances are not digested, and are not appreciably, if at all, reduced in bulk. Effects of the vapors of chloroform sulfuric ether and hydrocyanic acid. A plant bearing one leaf was introduced into a large bottle with a dram 3 Roman 1050 of chloroform, the mouth being imperfectly closed with cotton wool. The vapor caused in 1 Roman 1000 the lobes to begin moving at an imperceptibly slow rate, but in 3 Roman 1000 the spikes crossed, and the leaf was soon completely shut. The dose, however, was much too large, for in between 2 and 3 HRS the leaf appeared as if burnt and soon died. 2 leaves were exposed for 30 Roman 1000 in a 2 Rs vessel to the vapor of 30 minims 1 Roman 1050 of sulfuric ether. 1 leaf closed after a time, as did the other, whilst being removed from the vessel. Without being touched, both leaves were greatly injured. Another leaf, exposed for 20 Roman 1000 to 15 minims of ether, closed its lobes to a certain extent, and the sensitive filaments were now quite insensible. After 24 HRS, this leaf recovered its sensibility, but was still rather torpid. A leaf exposed in a large bottle for only 3 Roman 1000 to 10 drops was rendered insensible. After 52 Roman 1000, it recovered its sensibility, and when one of the filaments was touched, the lobes closed. It began page 305 to reopen. After 20 HRS, lastly, another leaf was exposed for 4 Roman 1000 to only 4 drops of the ether. It was rendered insensible, and did not close when its filaments were repeatedly touched, but closed when the end of the open leaf was cut off. This shows either that the internal parts had not been rendered insensible, or that an incision is a more powerful stimulus than repeated touches on the filaments. Whether the larger doses of chloroform and ether, which caused the leaves to close, slowly acted on the sensitive filaments, or on the leaf itself, I do not know. Cyanide of potassium, when left in a bottle, generates prussic or hydrocyanic acid. A leaf was exposed for 1 HR 35 Roman 1000 to the vapor thus formed, and the glands became within this time so colorless and shrunken as to be scarcely visible, and I at first thought that they had all dropped off. The leaf was not rendered insensible, for as soon as one of the filaments was touched, it closed. It had, however, suffered, for it did not reopen until nearly 2 days had passed, and was not even then in the least sensitive. After an additional day, it recovered its powers, and closed on being touched, and subsequently reopened. Another leaf behaved in nearly the same manner after a shorter exposure to this vapor. On the manner in which insects are caught, we will now consider the action of the leaves. When insects happen to touch one of the sensitive filaments, this often occurred in my greenhouse, but I do not know whether insects are attracted in any special way by the leaves. They are caught in large numbers by the plant in its native country. As soon as a filament is touched, both lobes close with astonishing quickness, and as they stand at less than a right angle to each other, they have a good chance of catching any intruder. The angle between the blade and footstalk does not change when the lobes close. The chief seat of movement is near the midrib, but is not confined to this part, for as the lobes come together, each curves inwards across its whole breadth, the marginal spikes, however, not becoming curved. This move, page 306, meant of the whole lobe was well seen in a leaf to which a large fly had been given, and from which a large portion had been cut off the end of one lobe, so that the opposite lobe meeting with no resistance in this. Part went on curving inwards much beyond the medial line. The whole of the lobe, from which a portion had been cut, was afterwards removed, and the opposite lobe now curled completely over, passing through an angle of from 120° to 130°, so as to occupy a position almost at right angles to that which it would have held had the opposite lobe been present. From the curving inwards of the two lobes, as they move towards each other, the straight marginal spikes intercross by their tips at first and ultimately by their bases. The leaf is then completely shut and encloses a shallow cavity if it has been made to shut merely by one of the sensitive filaments having been touched, or if it includes an object not yielding soluble nitrogenous matter. The two lobes retain their inwardly concave form until they re-expand. The re-expansion under these circumstances, that is when no organic matter is enclosed, was observed in 10 cases. In all of these, the leaves re-expanded to about 2/3 of the full extent in 24 HRS from the time of closure. Even the leaf, from which a portion of one lobe had been cut off, opened to a slight degree within the same time. In one case, a leaf re-expanded to about 2/3 of the full extent in 7 HRS and completely in 32 HRS, but one of its filaments had been touched merely with a hair, just enough to cause the leaf to close. Of these 10 leaves, only a few re-expanded completely in less than 2 days, and 2 or 3 required even a little longer time before. However they fully re-expand, they are ready to close. Page 307. Instantly, if their sensitive filaments are touched. How many times a leaf is capable of shutting and opening if no animal matter is left enclosed, I do not know, but one leaf was made to close 4 times, reopening afterwards within 6 days. On the last occasion, it caught a fly and then remained closed for many days. This power of reopening quickly after the filaments have been accidentally touched by blades of grass or by objects blown on the leaf by the wind, as occasionally happens in its native place, must be of some importance to the plant, for as long as a leaf remains closed it cannot of course capture an insect. When the filaments are irritated and a leaf is made to shut over an insect, a bit of meat, albumin, gelatin, casein, and no doubt any other substance containing soluble nitrogenous matter, the lobes, instead of remaining concave, thus including a concavity, slowly press closely together throughout their whole breadth. As this takes place, the margins gradually become a little everted, so that the spikes which at first intercrossed at last project in 2 parallel rows. The lobes press against each other with such force that I have seen a cube of albumin much flattened with distinct impressions of the little prominent glands. But this latter circumstance may have been partly caused by the corroding action of the secretion. So firmly do they become pressed together that if any large insect or other object has been caught, a corresponding projection on the outside of the leaf is distinctly visible. When the two lobes are thus completely shut, they, according to Dr. Curtis, in Boston Journal of Nat. Hist. Vol. I. 1837 p. 123, page 308, resist being opened as by a thin wedge driven between them with astonishing force, and are generally ruptured rather than yield. If not ruptured, they close again, as Dr. Canby informs me in a letter, with quite a loud flap. But if the end of a leaf is held firmly between the thumb and finger, or by a clip, so that the lobes cannot begin to close, they exert, whilst in this position, very little force. I thought at first that the gradual pressing together of the lobes was caused exclusively by captured insects crawling over and repeatedly irritating the sensitive filaments, and this view seemed the more probable when I learnt from Dr. Burden Sanderson that whenever the filaments of a closed leaf are irritated, the normal electric current is disturbed. Nevertheless, such irritation is by no means necessary for a dead insect or a bit of meat or of albumin. All act equally well, proving that in these cases it is the absorption of animal matter which excites the lobes slowly to press close together. We have seen that the absorption of an extremely small quantity of such matter also causes a fully expanded leaf to close slowly, and this movement is clearly analogous to the slow pressing together of the concave lobes. This latter action is of high functional importance to the plant, for the glands on both sides are thus brought into contact with a captured insect, and consequently secrete. The secretion with animal matter in solution is then drawn by capillary attraction over the whole surface of the leaf, causing all the glands to secrete and allowing them to absorb the diffused animal matter. The movement excited by the absorption of such matter, though slow, suffices for its final purpose, whilst the movement excited by one of the sensitive filaments being touched is rapid. And this is index page 309 pensible for the capturing of insects. These two movements excited by two such widely different means are thus both well adapted, like all the other functions of the plant, for the purposes which they subserve. There is another wide difference in the action of leaves, which enclose objects such as bits of wood, cork, balls of paper, or which have had their filaments merely touched, and those which enclose organic bodies yielding soluble nitrogenous matter. In the former case, the leaves, as we have seen, open in under 24 HRS, and are then ready even before being fully expended to shut again. But if they have closed over nitrogen-yielding bodies, they remain closely shut for many days, and after re-expanding are torpid, and never act again, or only after a considerable interval of time. In 4 instances, leaves, after catching insects, never reopened, but began to wither, remaining closed in one case for 15 days over a fly, in a second for 24 days, though the fly was small, in a third for 24 days over a woodlouse, and in a fourth for 35 days over a large tipula. In 2 other cases, leaves remained closed for at least 9 days over flies, and for how many more I do not know. It should, however, be added that in 2 instances, in which very small insects had been naturally caught, the leaf opened as quickly as if nothing had been caught, and I suppose that this was due to such small insects not having been crushed, or not having excreted any animal matter, so that the glands were not excited. Small angular bits of albumin and gelatine were placed at both ends of 3 leaves, 2 of which remained closed for 13, and the other for 12 days. 2 other leaves remained closed over bits of page 310 meat for 11 days, a third leaf for 8 days, and a fourth, but this had been cracked and injured, for only 6 days. Bits of cheese or casein were placed at one end and albumin at the other end of 3 leaves, and the ends with the former opened after 6, 8, and 9 days, whilst the opposite ends opened a little later. None of the above bits of meat, albumin Roman 100, exceeded a cube of 110 of an inch 2 Roman 2000 in size, and were sometimes smaller, yet these small portions sufficed to keep the leaves closed for many days. Dr. Canby informs me that leaves remain shut for a longer time over insects than over meat, and from what I have seen, I can well believe that this is the case, especially if the insects are large. In all the above cases, and in many others in which leaves remained closed for a long but unknown period over insects naturally caught, they were more or less torpid. When they reopened, generally, they were so torpid during many succeeding days that no excitement of the filaments caused the least movement. In one instance, however, on the day after a leaf opened, which had clasped a fly, it closed with extreme slowness when one of its filaments was touched, and although no object was left enclosed, it was so torpid that it did not reopen for the second time until 44 HRS had elapsed. In a second case, a leaf, which had expanded after remaining closed for at least 9 days over a fly, when greatly irritated, moved one alone of its 2 lobes, and retained this unusual position for the next 2 days. A third case offers the strongest exception which I have observed. A leaf, after remaining clasped for an unknown time over a fly, opened, and when one of its filaments was touched, closed, though rather slowly. Dr. Canby, page 311, who observed in the United States a large number of plants, which, although not in their native site, were probably more vigorous than my plants, informs me that he has several times known vigorous leaves to devour their prey several times, but ordinarily twice, or quite often once, was enough to render them unserviceable. Mrs. Treat, who cultivated many plants in New Jersey, also informs me that several leaves caught successively 3 insects each, but most of them were not able to digest the third fly, but died in the attempt. 5 leaves, however, digested each 3 flies and closed over the fourth, but died soon after the fourth capture. Many leaves did not digest even one large insect. It thus appears that the power of digestion is somewhat limited, and it is certain that leaves always remain clasped for many days over an insect, and do not recover their power of closing again for many subsequent days. In this respect, Dionea differs from Drosera, which catches and digests many insects after shorter intervals of time. We are now prepared to understand the use of the marginal spikes, which form so conspicuous a feature in the appearance of the plant Fig. 12p. 287, and which at first seemed to me in my ignorance useless appendages from the inward curvature of the lobes. As they approach each other, the tips of the marginal spikes first intercross, and ultimately their bases, until the edges of the lobes come into contact, elongated spaces between the spikes, varying from the 115 to the 110 of an inch 1 to 2 Roman 2000 in breadth, according to the size of the leaf, are left open. Thus an insect, if its body is not thicker than these measurements, can easily escape between the crossed spikes when disturbed by the closing lobes, and in page 312 creasing darkness, and one of my sons actually saw a small insect thus escaping. A moderately large insect, on the other hand, if it tries to escape between the bars, will surely be pushed back again into its horrid prison, with closing walls, for the spikes continue to cross more and more, until the edges of the lobes come into contact. A very strong insect, however, would be able to free itself, and Mrs. Treat saw this effected by a rosechafer Macrodactylus subspinosus in the United States. Now, it would manifestly be a great disadvantage to the plant to waste many days in remaining clasped over a minute insect, and several additional days or weeks in afterwards recovering its sensibility. Inasmuch as a minute insect would afford but little nutriment, it would be far better for the plant to wait for a time until a moderately large insect was captured, and to allow all the little ones to escape, and this advantage is secured by the slowly intercrossing marginal spikes, which act like the large meshes of a fishing net, allowing the small and useless fry to escape. As I was anxious to know whether this view was correct, and as it seems a good illustration of how cautious we ought to be in assuming, as I had done with respect to the marginal spikes, that any fully developed structure is useless, I applied to Dr. Canby. He visited the native site of the plant early in the season, before the leaves had grown to their full size, sent me 14 leaves containing naturally captured insects. 4 of these had caught rather small insects, viz. 3 of them ants, and the 4th a rather small fly, but the other 10 had all caught large insects, namely 5 elaters, 2 chrysomlas, a curculio, a thick and broad spider, and a scolopendra. Out of these 10 insects, no less than 8, page 313, were beetles, and out of the whole 14, there was only one, viz. A dipterous insect, which could readily take flight. Drosera, on the other hand, lives chiefly on insects, which are good flyers, especially Diptera, caught by the aid of its viscid secretion. But what most concerns us is the size of the 10 larger insects. Their average length from head to tail was 256 of an inch, the lobes of the leaves being on an average 53 of an inch in length, so that the insects were very nearly half as long as the leaves within which they were enclosed. Only a few of these leaves, therefore, had wasted their powers by capturing small prey, though it is probable that many small insects had crawled over them and been caught, but had then escaped through the bars. The transmission of the motor impulse and means of movement is sufficient to touch any one of the 6 filaments to cause both lobes to close, these becoming at the same time incurved throughout their whole breadth. The stimulus must therefore radiate in all directions from any one filament. It must also be transmitted with much rapidity across the leaf, for in all ordinary cases, both lobes close simultaneously as far as the eye can judge. Most physiologists believe that in irritable plants, the excitement is transmitted along or in close connection with the fibrovascular bundles in Dionea, the course of these vessels composed of spiral and ordinary vascular. Dr. Canby remarks Gardner's Monthly, August 1868, as a general thing, beetles and insects of that kind, though always killed, seem to be too hard-shelled to serve as food, and after a short time are rejected. I am surprised at this statement, at least with respect to such beetles as elaters, for the 5 which I examined were in an extremely fragile and empty condition, as if all their internal parts had been partially digested. Mrs. Treat informs me that the plants which she cultivated in New Jersey chiefly caught Diptera, page 314. Tissue seems at first sight to favor this belief, for they run up the midrib in a great bundle, sending off small bundles almost at right angles on each side. These bifurcate occasionally as they extend towards the margin, and close to the margin small branches from adjoining vessels unite, and enter the marginal spikes at some of these points of union. The vessels form curious loops, like those described under Drosera. A continuous zigzag line of vessels thus runs round the whole circumference of the leaf, and in the midrib, all the vessels are in close contact, so that all parts of the leaf seem to be brought into some degree of communication. Nevertheless, the presence of vessels is not necessary for the transmission of the motor impulse, for it is transmitted from the tips of the sensitive filaments, these being about the 120th of an inch in length, into which no vessels enter, and these could not have been overlooked, as I made thin vertical sections of the leaf at the bases of the filaments. On several occasions, slits about the 110th of an inch in length were made with a lancet close to the bases of the filaments parallel to the midrib, and therefore directly across the course of the vessels. These were made sometimes on the inner and sometimes on the outer sides of the filaments, and after several days, when the leaves had reopened, these filaments were touched roughly, for they were always rendered, in some degree, torpid by the operation, and the lobes then closed in the ordinary manner, though slowly and sometimes not, until after a considerable interval of time. These cases show that the motor impulse is not transmitted along the vessels, and they further show that there is no necessity for a direct line of communication from the filament, which is page 315, touched towards the midrib and opposite lobe, or towards the outer parts of the same lobe. Two slits near each other, both parallel to the midrib, were next made in the same manner as before, one on each side of the base of a filament on 5 distinct leaves, so that a little slip bearing a filament was connected with the rest of the leaf only at its 2 ends. These slips were nearly of the same size. One was carefully measured. It was 12 of an inch 3 Roman 2000 in length, and 8 of an inch 2 Roman 2000 in breadth, and in the middle stood the filament. Only one of these slips withered and perished after the leaf had recovered from the operation. Though the slits were still open, the filaments thus circumstanced were roughly touched, and both lobes, or one alone, slowly closed. In 2 instances, touching the filament produced no effect, but when the point of a needle was driven into the slip at the base of the filament, the lobes slowly closed. Now, in these cases, the impulse must have proceeded along the slip in a line parallel to the midrib, and then have radiated forth either from both ends or from one end alone of the slip over the whole surface of the 2 lobes. Again, 2 parallel slits like the former ones were made, one on each side of the base of a filament at right angles to the midrib. After the leaves, 2 in number, had recovered, the filaments were roughly touched, and the lobes slowly closed, and here the impulse must have traveled for a short distance in a line at right angles to the midrib, and then have radiated forth on all sides over both lobes. These several cases prove that the motor impulse travels in all directions through the cellular tissue, independently of the course of the vessels. With Drosera, we have seen that the motor impulse, page 316, is transmitted in like manner in all directions through the cellular tissue, but that its rate is largely governed by the length of the cells and the direction of their longer axes. Thin sections of a leaf of Dionea were made by my son, and the cells, both those of the central and of the more superficial layers, were found much elongated with their longer axes directed towards the midrib, and it is in this direction that the motor impulse must be sent with great rapidity from one lobe to the other, as both close simultaneously. The central parenchymatous cells are larger, more loosely attached together, and have more delicate walls than the more superficial cells. A thick mass of cellular tissue forms the upper surface of the midrib over the great central bundle of vessels. When the filaments were roughly touched at the bases of which slits had been made either on both sides or on one side parallel to the midrib or at right angles to it, the 2 lobes or only one moved. In one of these cases, the lobe on the side which bore the filament that was touched moved, but in 3 other cases, the opposite lobe alone moved, so that an injury which was sufficient to prevent a lobe moving did not prevent the transmission from it of a stimulus which excited the opposite lobe to move. We thus also learn that although normally both lobes move together, each has the power of independent movement. A case, indeed, has already been given of a torpid leaf that had lately reopened after catching an insect of which one lobe alone moved. When irritated, moreover, one end of the same lobe can close and re-expand independently of the other end, as was seen in some of the foregoing experiments. When the lobes, which are rather thick, close, no trace of wrinkling, can be seen on any part of their upper page 317 surfaces, it appears therefore that the cells must contract. The chief seat of the movement is evidently in the thick mass of cells, which overlies the central bundle of vessels in the midrib. To ascertain whether this part contracts, a leaf was fastened on the stage of the microscope in such a manner that the two lobes could not become quite shut. And having made 2-minute black dots on the midrib in a transverse line and a little towards one side, they were found by the micrometer to be 171,000 of an inch apart. One of the filaments was then touched, and the lobes closed, but as they were prevented from meeting, I could still see the two dots, which now were 151,000 of an inch apart, so that a small portion of the upper surface of the midrib had contracted in a transverse line 21,000 of an inch, 508 Roman 2000. We know that the lobes, whilst closing, become slightly incurved throughout their whole breadth. This movement appears to be due to the contraction of the superficial layers of cells over the whole upper surface. In order to observe their contraction, a narrow strip was cut out of one lobe at right angles to the midrib, so that the surface of the opposite lobe could be seen in this part when the leaf was shut after the leaf had recovered from the operation and had re-expanded. 3-minute black dots were made on the surface opposite to the slit or window in a line at right angles to the midrib. The distance between the dots was found to be 401,000 of an inch, so that the two extreme dots were 801,000 of an inch apart. One of the filaments was now touched, and the leaf closed on again. Measuring the distances between the dots, the two next to the midrib were nearer together by 1 to 21,000 of an inch, and the two further dots by 3 to 41,000 of an inch than they were before, so that the two extreme page 318 dots now stood about 51,000 of an inch, 127 Roman 2000, nearer together than before. If we suppose the whole upper surface of the lobe, which was 4,001,000 of an inch in breadth, to have contracted in the same proportion, the total contraction will have amounted to about 251,000 or 140 of an inch, 635 Roman 2000. But whether this is sufficient to account for the slight inward curvature of the whole lobe, I am unable to say. Finally, with respect to the movement of the leaves, the wonderful discovery made by Dr. Burden Sanderson is now universally known, namely, that there exists a normal electrical current in the blade and footstalk, and that when the leaves are irritated, the current is disturbed in the same manner as takes place during the contraction of the muscle of an animal. The re-expansion of the leaves, this, is effected at an insensibly slow rate. Whether or not any object is enclosed, one lobe can re-expand by itself, as occurred with the torpid leaf, of which one lobe alone had closed. We have also seen in the experiments with cheese and albumin that the two ends of the same lobe can re-expand to a certain extent independently of each other. But in all ordinary cases, both lobes open at the same time. The re-expansion is not determined by the sensitive filaments. All three filaments on one lobe were cut off close to their bases, and the three. Proc. Royal Soc. Vol. Roman 21 p. 495, and Lecture at the Royal Institution, June 5, 1874, given in Nature 1874, pp. 105 and 127. Not all in his Gen. American Plants p. 277 note says that whilst collecting this plant in its native home, I had occasion to observe that a detached leaf would make repeated efforts towards disclosing itself to the influence of the sun. These attempts consisted in an undulatory motion of the marginal cilii, accompanied by a partial opening and succeeding collapse of the lamina, which at length terminated in a complete expansion. And in the destruction of sensibility, I am indebted to Prof. Oliver for this reference, but I do not understand what took place. Page 319. Leaves thus treated re-expanded on to a partial extent in 24 HRSA second, to the same extent in 48 HRS, and the third, which had been previously injured, not until the 6th day. These leaves, after their re-expansion, closed quickly. When the filaments on the other lobe were irritated, these were then cut off one of the leaves, so that none were left. This mutilated leaf, notwithstanding the loss of all its filaments, re-expanded in 2 days in the usual manner, when the filaments have been excited by immersion in a solution of sugar, the lobes do not expand so soon as when the filaments have been merely touched, and this, I presume, is due to their having been strongly affected through exosmose, so that they continue for some time to transmit a motor impulse to the upper surface of the leaf. The following facts make me believe that the several layers of cells forming the lower surface of the leaf are always in a state of tension, and that it is owing to this mechanical state, aided probably by fresh fluid being attracted into the cells, that the lobes begin to separate or expand as soon as the contraction of the upper surface diminishes. A leaf was cut off and suddenly plunged perpendicularly into boiling water. I expected that the lobes would have closed, but instead of doing so, they diverged a little. I then took another fine leaf, with the lobes standing at an angle of nearly 80° to each other, and on immersing it, as before, the angle suddenly increased to 90°. A third leaf was torpid from having recently re-expanded after having caught a fly, so that repeated touches of the filaments caused not the least movement. Nevertheless, when similarly immersed, the lobes separated a little, as these leaves were inserted perpendicularly into the boiling water. Both surfaces and the filaments, page 320, must have been equally affected, and I can understand the divergence of the lobes only by supposing that the cells on the lower side, owing to their state of tension, acted mechanically, and thus suddenly drew the lobes a little apart as soon as the cells on the upper surface were killed and lost their contractile power. We have seen that boiling water in like manner causes the tentacles of Drosera to curve backwards, and this is an analogous movement to the divergence of the lobes of Dionea. In some concluding remarks in the 15th chapter on the Droseraceae, the different kinds of irritability possessed by the several genera, and the different manner in which they capture insects, will be compared, page 321. Chapter Roman 14, Aldrovanda vesiculosa, captures crustacean structure of the leaves in comparison with those of Dionea. Absorption by the glands, by the quadruped processes, and points on the enfolded margins. Aldrovanda vesiculosa, var. Australis, captures prey absorption of animal matter. Aldrovanda vesiculosa, var. verticillata. Concluding remarks. This plant may be called a miniature aquatic Dionea Stein, discovered in 1873, that the bilobed leaves, which are generally found closed in Europe, open under a sufficiently high temperature, and when touched suddenly close they re-expand in from 24 to 36 hours, but only as it appears when inorganic objects are enclosed. The leaves sometimes contain bubbles of air and were formerly supposed to be bladders, hence the specific name of vesiculosa. Stein observed that water insects were sometimes caught, and Prof. Cohn has recently found within the leaves of naturally growing plants many kinds of crustaceans and larvae. Plants which had been kept in filtered water were placed by him in a vessel can. Since his original publication, Stein has found out that the irritability of the leaves was observed by de Saussure, as recorded in Bull Bot Soche de France in 1861. Del Pino states in a paper published in 1871, Nuovo Giornale Bot Ital Val Roman 3 p. 174, that una quantit di chioxialine e di oltre animalculi aquatici are caught and suffocated by the leaves. I presume that chioxialine are freshwater mollusks. It would be interesting to know whether their shells are at all corroded by the acid of the digestive secretion. I am greatly indebted to this distinguished naturalist for having sent me a copy of his memoir on Aldrovanda before its publication in his Beiträge zur Biologie der Pflanzen Dritts Heft, 1875, page 71, page 322. Containing numerous crustaceans of the genus Cypris, and next morning many were found imprisoned and alive, still swimming about within the closed leaves, but doomed to certain death. Directly after reading Prof. Cohn's memoir, I received through the kindness of Dr. Hooker living plants from Germany. As I can add nothing to Prof. Cohn's excellent description, I will give only two illustrations, one of a wall of leaves copied from his work, and the other of a leaf pressed flat open, drawn by my son Francis. I will, however, append a few remarks on the differences between this plant and Dionea. Aldrovanda is destitute of roots and floats freely in the water, The leaves are arranged in whorls round the stem. Their broad petioles terminate in from 4 to 6 rigid projections, each tipped with a stiff, short bristle. The bilobed leaf, with the midrib likewise tipped with a bristle, stands in the midst of these projections, and is evidently defended by them. The lobes are formed of very delicate tissue, so as to be translucent. They open, according to Cohn, about as much as the 2 valves of a living mussel shell, therefore even less than the lobes of Dionea, and this must make the capture of aquatic animals more easy. The outside of the leaves and the petioles are covered with minute 2-armed papillae, evidently answering to the atrade papillae of Dionea. Each lobe rather exceeds a semicircle in convexity, and consists of 2 very different concentric portions, the inner and lesser portion, or that next to the midrib. There has been much discussion by botanists on the homological nature of these projections. Dr. Nitschke Bot, Zeitung, 1861, p. 146, believes that they correspond with the fimbriated scale-like bodies found at the bases of the petioles of Drosera, page 323. Is slightly concave, and is formed, according to Cohn, of 3 layers of cells. Its upper surface is studded with colorless glands like, but more simple than those of Dionea. They are supported on distinct footstalks, consisting of 2 rows of cells. The outer. Fig. 13, Aldrovanda vesiculosa, upper figure whorl of leaves, from Prof. Cohn. Lower figure leaf, pressed flat open and greatly enlarged. And broader portion of the lobe is flat and very thin, being formed of only 2 layers of cells. Its upper surface does not bear any glands, but in their place small quadruped processes, each consisting of 4 tapering projections, which rise from a common, page 324, prominence. These processes are formed of very delicate membrane lined with a layer of protoplasm, and they sometimes contain aggregated globules of hyaline matter. 2 of the slightly diverging arms are directed towards the circumference, and 2 towards the midrib, forming together a sort of Greek cross. Occasionally 2 of the arms are replaced by 1, and then the projection is trifid. We shall see in a future chapter that these projections curiously resemble those found within the bladders of Utricularia, more especially of Utricularia montana, although this genus is not related to Aldrovanda. A narrow rim of the broad, flat exterior part of each lobe is turned inwards, so that when the lobes are closed, the exterior surfaces of the enfolded portions come into contact. The edge itself bears a row of conical, flattened, transparent points with broad bases like the prickles on the stem of a bramble or rubus. As the rim is enfolded, these points are directed towards the midrib, and they appear at first as if they were adapted to prevent the escape of prey. But this can hardly be their chief function, for they are composed of very delicate and highly flexible membrane, which can be easily bent or quite doubled back without being cracked. Nevertheless, the enfolded rims, together with the points, must somewhat interfere with the retrograde movement of any small creature. As soon as the lobes begin to close, the circumferential part of the leaf of Aldrovanda thus differs greatly from that of Dionea, nor can the points on the rim be considered as homologous with the spikes round the leaves of Dionea, as these latter are prolongations of the blade, and not mere epidermic productions. They appear also to serve for a widely different purpose, page 325. On the concave gland-bearing portion of the lobes, and especially on the midrib, there are numerous long, finely pointed hairs, which, as Prof. Cohn remarks, there can be little doubt, are sensitive to a touch, and when touched cause the leaf to close. They are formed of 2 rows of cells, or, according to Cohn, sometimes of 4, and do not include any vascular tissue. They differ also from the 6 sensitive filaments of Dionea in being colorless, and in having a medial as well as a basal articulation. No doubt it is owing to these 2 articulations that, notwithstanding their length, they escape being broken when the lobes close. The plants which I received during the early part of October from Kew never opened their leaves, though subjected to a high temperature. After examining the structure of some of them, I experimented on only 2, as I hoped that the plants would grow, and I now regret that I did not sacrifice a greater number. A leaf was cut open along the midrib, and the glands examined under a high power. It was then placed in a few drops of an infusion of raw meat. After 3 HRS 20 Roman 1000, there was no change, but when next examined after 23 HRS 20 Roman 1000, the outer cells of the glands contained, instead of limpid fluid, spherical masses of a granular substance, showing that matter had been absorbed from the infusion, that these glands secrete a fluid which dissolves or digests animal matter out of the bodies of the creatures, which the leaves capture, is also highly probable from the analogy of Dionea, if we may trust to the same analogy, the concave and inner portions of the two lobes probably close together by a slow movement. As soon as the glands have absorbed a slight amount of page 326 already soluble animal matter, the included water would thus be pressed out, and the secretion consequently not be too much diluted to act with respect to the quadruped processes on the outer parts of the lobes. I was not able to decide whether they had been acted on by the infusion, for the lining of protoplasm was somewhat shrunk before they were immersed. Many of the points on the enfolded rims also had their lining of protoplasm similarly shrunk and contained spherical granules of hyaline matter. A solution of urea was next employed. This substance was chosen partly because it is absorbed by the quadruped processes, and more especially by the glands of utricularia plant, which, as we shall hereafter see, feeds on decayed animal matter, as urea is one of the last products of the chemical changes going on in the living body. It seems fitted to represent the early stages of the decay of the dead body. I was also led to try urea from a curious little fact mentioned by Prof. Cohn, namely, that when rather large crustaceans are caught between the closing lobes, they are pressed so hard whilst making their escape that they often void their sausage-shaped masses of excrement, which were found within most of the leaves. These masses no doubt contain urea. They would be left either on the broad, outer surfaces of the lobes, where the quadrupeds are situated, or within the closed concavity, in the latter case water charged with excrementitious and decaying matter would be slowly forced outwards, and would bathe the quadrupeds. If I am right in believing that the concave lobes contract after a time like those of Dionea, foul water would also be apt to ooze out at all times, especially when bubbles of air were generated within the concavity. A leaf was cut open and examined, and the outer page 327 cells of the glands were found to contain only limpid fluid. Some of the quadrupeds included a few spherical granules, but several were transparent and empty, and their positions were marked. This leaf was now immersed in a little solution of 1 part of urea to 146 of water, or 3 grains to the ounce, after 3 HRS 40 Roman 1000. There was no change either in the glands or quadrupeds, nor was there any certain change in the glands after 24 HRS, so that as far as one trial goes, urea does not act on them in the same manner as an infusion of raw meat. It was different with the quadrupeds, for the lining of protoplasm, instead of presenting a uniform texture, was now slightly shrunk and exhibited, in many places, minute, thickened, irregular yellowish specks and ridges exactly like those which appear within the quadrupeds of utricularia when treated with this same solution. Moreover, several of the quadrupeds which were before empty now contain moderately sized or very small, more or less aggregated globules of yellowish matter, as likewise occurs under the same circumstances with utricularia. Some of the points on the enfolded margins of the lobes were similarly affected, for their lining of protoplasm was a little shrunk and included yellowish specks, and those which were before empty now contained small spheres and irregular masses of hyaline matter, more or less aggregated, so that both the points on the margins and the quadrupeds had absorbed matter from the solution in the course of 24 HRS. But to this subject I shall recur in another, rather old leaf, to which nothing had been given, but which had been kept in foul water. Some of the quadrupeds contained aggregated translucent globules. These were not acted on by a solution of 1 part of carbonate of ammonia to 218 of water, and this negative result, page 328, agrees with what I have observed under similar circumstances with utricularia. Aldrovanda vesiculosa var australis. Dried leaves of this plant from Queensland in Australia were sent me by Prof. Oliver from the herbarium at Kew. Whether it ought to be considered as a distinct species or a variety cannot be told until the flowers are examined by a botanist The projections at the upper end of the petiole, from 4 to 6 in number, are considerably longer relatively to the blade, and much more attenuated than those of the European form. They are thickly covered for a considerable space near their extremities with the upcurved prickles, which are quite absent in the latter form, and they generally bear on their tips 2 or 3 straight prickles instead of one. The bilobed leaf appears also to be rather larger and somewhat broader, with the pedicel, by which it is attached to the upper end of the petiole, a little longer. The points on the enfolded margins likewise differ. They have narrower bases, and are more pointed. Long and short points also alternate with much more regularity than in the European form. The glands and sensitive hairs are similar in the two forms. No quadruped processes could be seen on several of the leaves, but I do not doubt that they were present, though indistinguishable from their delicacy, and from having shriveled, for they were quite distinct on one leaf under circumstances presently to be mentioned. Some of the closed leaves contain no prey, but in one there was a rather large beetle, which from its flattened tibiae I suppose was an aquatic species, but was not allied to colymbetes. All the softer tissues of this beetle were completely dissolved, and its chitinous integuments were as clean as if they had been page 329 boiled in caustic potash, so that it must have been enclosed for a considerable time. The glands were browner and more opaque than those on other leaves which had caught nothing, and the quadruped processes, from being partly filled with brown granular matter, could be plainly distinguished, which was not the case as already stated on the other leaves. Some of the points on the enfolded margins likewise contained brownish granular matter. We thus gain additional evidence that the glands, the quadruped processes, and the marginal points all have the power of absorbing matter, though probably of a different nature. Within another leaf, disintegrated remnants of a rather small animal, not a crustacean, which had simple, strong, opaque mandibles, and a large, unarticulated chitinous coat, were present. Lumps of black organic matter, possibly of a vegetable nature, were enclosed in 2 other leaves, but in one of these there was also a small worm much decayed. But the nature of partially digested and decayed bodies, which have been pressed flat, long dried, and then soaked in water, cannot be recognized easily. All the leaves contained unicellular and other algae still of a greenish color, which had evidently lived as intruders in the same manner as occurs according to Cohn within the leaves of this plant in Germany. Aldrovanda vesiculosa var verticillata. Dr. King, superintendent of the Botanic Gardens, kindly sent me dried specimens collected near Calcutta. This form was, I believe, considered by Wallach as a distinct species under the name of verticillata. It resembles the Australian form much more nearly than the European, namely in the projections at the upper end of the petiole being much attenuated and covered with page 330 upcurved prickles. They terminate also in 2 straight little prickles. The bilobed leaves are, I believe, larger and certainly broader even than those of the Australian form, so that the greater convexity of their margins was conspicuous, the length of an open leaf being taken at 100. The breadth of the Bengal form is nearly 173, of the Australian form 147, and of the German 134. The points on the enfolded margins are like those in the Australian form. Of the few leaves which were examined, 3 contained entomostracan crustaceans. Concluding remarks: the leaves of the 3 foregoing closely allied species or varieties are manifestly adapted for catching living creatures. With respect to the functions of the several parts, there can be little doubt that the long jointed hairs are sensitive like those of Dionea, and that when touched they cause the lobes to close, that the glands secrete a true digestive fluid and afterwards absorb the digested matter, is highly probable from the analogy of Dionea, from the limpid fluid within their cells being aggregated into spherical masses after they had absorbed an infusion of raw meat from their opaque and granular condition in the leaf, which had enclosed a beetle for a long time, and from the clean condition of the integuments of this insect, as well as of crustaceans as described by Cohn, which have been long captured again from the effect produced on the quadruped processes by an immersion for 24 HRS in a solution of urea, from the presence of brown granular matter within the quadrupeds of the leaf in which the beetle had been caught, and from the analogy of utriculariite, is probable that these processes absorb excrementitious and decaying animal matter, it is a more curious fact that the points on page 331, the enfolded margins, apparently serve to absorb decayed animal matter in the same manner as the quadrupeds. We can thus understand the meaning of the enfolded margins of the lobes, furnished with delicate points directed inwards, and of the broad, flat outer portions. Bearing quadruped processes for these surfaces must be liable to be irrigated by foul water flowing from the concavity of the leaf. When it contains dead animals, this would follow from various causes, from the gradual contraction of the concavitiform fluid in excess being secreted, and from the generation of bubbles of air. More observations are requisite on this head, but if this view is correct, we have the remarkable case of different parts of the same leaf serving for very different purpose, sown part for true digestion, and another for the absorption of decayed animal matter. We can thus also understand how by the gradual loss of either power, a plant might be gradually adapted for the one function to the exclusion of the other. And it will hereafter be shown that two genera, namely Pinguicula and Utricularia, belonging to the same family, have been adapted for these two different functions, page 332. Chapter Roman 15, Drosophilum roridula biblis glandula hairs of other plants, concluding remarks on the Drose race. Drosophilum, structure of leaves, nature of the secretion, manner of catching insects, power of absorption, digestion of animal substances. Summary on Drosophilum roridula biblis glandular hairs of other plants, their power of absorption. Saxifraga, Primula, Pelargonium, Erica, Mirabilis nicotiana. Summary on glandular hairs, concluding remarks on the Drose race. Drosophilum lusitanicum, this rare plant has been found only in Portugal, and as I hear from Dr. Hooker in Morocco, I obtained living specimens through the great kindness of Mr. W. Roman 100 Tate, and afterwards from Mr. G. Maw and Dr. Moore. Mr. Tate informs me that it grows plentifully on the sides of dry hills near Oporto, and that vast numbers of flies adhere to the leaves. This latter fact is well known to the villagers, who call the plant the flycatcher and hang it up in their cottages for this purpose. A plant in my hothouse caught so many insects during the early part of April, although the weather was cold and insects scarce, that it must have been in some manner strongly attractive to them. On 4 leaves of a young and small plant, 8, 10, 14, and 16-minute insects, chiefly Diptera, were found in the autumn adhering to them. I neglected to examine the roots, but I hear from Dr. Hooker that they are very small, as in the case of the previously mentioned members of the same family of the Drose race. The leaves arise from an almost woody axis. They, page 333, are linear, much attenuated towards their tips, and several inches in length. The upper surface is concave, the lower convex, with a narrow channel down the middle. Both surfaces, with the exception of the channel, are covered with glands, supported on pedicels, and arranged in irregular longitudinal rows. These organs I shall call tentacles, from their close resemblance to those of Drosera, though they have no power of movement. Those on the same leaf differ much in length. The glands also differ in size, and are of a bright pink or of a purple color. Their upper surfaces are convex, and the lower flat or even concave, so that they resemble miniature mushrooms in appearance. They are formed of 2, as I believe, layers of delicate angular cells, enclosing 8 or 10 larger cells, with thicker zigzag walls within these larger cells. There are others marked by spiral lines, and apparently connected with the spiral vessels, which run up the green multicellular pedicels. The glands secrete large drops of viscid secretion. Other glands, having the same general appearance, are found on the flower peduncles and calyx. Fig 14, Drosophilum lusitanicum, part of leaf, enlarged 7 times, showing lower surface. Besides the glands, which are borne on longer or shorter pedicels, there are numerous ones both on the upper and lower surfaces of the leaves, so small as to be scarcely visible to the naked eye. They are colorless and almost sessile, either circular or oval in outline, the latter occurring chiefly on the backs of the leaves. Fig 14. Internally, they have exactly the same structure as the larger glands, which are supported on pedicels, page 334. and indeed the two sets almost graduate into one another, but the sessile glands differ in one important respect, for they never secrete spontaneously as far as I have seen, though I have examined them under a high power on a hot day, whilst the glands on pedicels were secreting copiously nevertheless. If little bits of damp albumin or fibrin are placed on these sessile glands, they begin after a time to secrete in the same manner as do the glands of Dionea when similarly treated when they were merely rubbed with a bit of raw meat. I believe that they likewise secreted. Both the sessile glands and the taller ones on pedicels have the power of rapidly absorbing nitrogenous matter. The secretion from the taller glands differs in a remarkable manner from that of Drosera, in being acid before the glands have been in any way excited, and judging from the changed color of litmus paper, more strongly acid than that of Drosera. This fact was observed repeatedly. On one occasion, I chose a young leaf, which was not secreting freely, and had never caught an insect, yet the secretion on all the glands colored litmus paper of a bright red from the quickness with which the glands are able to obtain animal matter from such substances as well-washed fibrin and cartilage. I suspect that a small quantity of the proper ferment must be present in the secretion before the glands are excited, so that a little animal matter is quickly dissolved. Owing to the nature of the secretion, or to the shape of the glands, the drops are removed from them with singular facility. It is even somewhat difficult, by the aid of a finely pointed polished needle, slightly damped with water, to place a minute particle of any kind on one of the drops, for on withdrawing the page 335 needle, the drop is generally withdrawn, whereas with Drosera there is no such difficulty, though the drops are occasionally withdrawn. From this peculiarity, when a small insect alights on a leaf of Drosophilum, the drops adhere to its wings, feet, or body, and are drawn from the gland. The insect then crawls onward, and other drops adhere to it, so that at last, bathed by the viscid secretion, it sinks down and dies, resting on the small sessile glands with which the surface of the leaf is thickly covered. In the case of Drosera, an insect sticking to one or more of the exterior glands is carried by their movement to the center of the leaf. With Drosophilum, this is effected by the crawling of the insect, as from its wings being clogged by the secretion it cannot fly away. There is another difference in function between the glands of these two plants. We know that the glands of Drosera secrete more copiously when properly excited, but when minute particles of carbonate of ammonia, drops of a solution of this salt, or of the nitrate of ammonia, saliva, small insects, bits of raw or roast meat, albumin, fibrin, or cartilage, as well as inorganic particles, were placed on the glands of Drosophilum, the amount of secretion never appeared to be in the least increased, as insects do not commonly adhere to the taller glands, but withdraw the secretion. We can see that there would be little use in their having acquired the habit of secreting copiously when stimulated, whereas with Drosera this is of use, and the habit has been acquired nevertheless. The glands of Drosophilum, without being stimulated, continually secrete, so as to replace the loss by evaporation. Thus, when a plant was placed under a small bell-glass, with its inner surface and support thoroughly wetted, there was no loss by evaporation, and so much page 336 secretion was accumulated in the course of a day that it ran down the tentacles and covered large spaces of the leaves. The glands to which the above-named nitrogenous substances and liquids were given did not, as just stated, secrete more copiously. On the contrary, they absorbed their own drops of secretion with surprising quickness. Bits of damp fibrin were placed on 5 glands, and when they were looked at after an interval of 1 hr 12 Roman 1000, the fibrin was almost dry, the secretion having been all absorbed. So it was with 3 cubes of albumin after 1 hr 19 Roman 1000, and with 4 other cubes, though these latter were not looked at until 2 hrs 15 Roman 1000, had elapsed. The same result followed in between 1 hr 15 Roman 1000 and 1 hr 30 Roman 1000, when particles both of cartilage and meat were placed on several glands. Lastly, a minute drop, about 120 of a minim, of a solution of 1 part of nitrate of ammonia to 146 of water, was distributed between the secretions surrounding 3 glands so that the amount of fluid surrounding each was slightly increased, yet when looked at after 2 HRS, all 3 were dry. On the other hand, 7 particles of glass and 3 of coal cinders of nearly the same size as those of the above-named organic substances were placed on 10 glands, some of them being observed for 18 HRS, and others for 2 or 3 days, but there was not the least sign of the secretion being absorbed. Hence, in the former cases, the absorption of the secretion must have been due to the presence of some nitrogenous matter, which was either already soluble or was rendered so by the secretion, as the fibrin was pure and had been well washed in distilled water after being kept in glycerine, and as the cartilage had been soaked in water. I suspect that these substances must, page 337, have been slightly acted on and rendered soluble within the above stated short periods. The glands have not only the power of rapid absorption, but likewise of secreting again quickly, and this latter habit has perhaps been gained inasmuch as insects, if they touch the glands, generally withdraw the drops of secretion which have to be restored. The exact period of resecretion was recorded in only a few cases. The glands on which bits of meat were placed, and which were nearly dry after about 1 HR 30 Roman 1000, when looked at after 22 additional hours, were found secreting. So it was after 24 HRS with 1 gland on which a bit of albumin had been placed, the 3 glands to which a minute drop of a solution of nitrate of ammonia was distributed, and which became dry after 2 HRS, were beginning to resecrete after only 12 additional hours. Tentacles incapable of movement. Many of the tall tentacles with insects adhering to them were carefully observed, and fragments of insects, bits of raw meat, albumin Roman 100 drops of a solution of 2 salts of ammonia and of saliva were placed on the glands of many tentacles, but not a trace of movement could ever be detected. I also repeatedly irritated the glands with a needle and scratched and pricked the blades, but neither the blade nor the tentacles became at all inflected. We may therefore conclude that they are incapable of movement. On the power of absorption possessed by the glands, it has already been indirectly shown that the glands on pedicels absorb animal matter, and this is further shown by their changed color and by the aggregation of their contents after they have been left in contact with nitrogenous substances or liquids. The following observations apply both to the glands supported on page 338 pedicels and to the minute sessile ones before a gland has been in any way stimulated. The exterior cells commonly contain only limpid purple fluid. The more central ones, including mulberry-like masses of purple granular matter. A leaf was placed in a little solution of 1 part of carbonate of ammonia to 146 of water, 3 GRS to 1 Rs, and the glands were instantly darkened and very soon became black, this change being due to the strongly marked aggregation of their contents, more especially of the inner cells. Another leaf was placed in a solution of the same strength of nitrate of ammonia, and the glands were slightly darkened in 25 Roman 1000, more so in 50 Roman 1000, and after 1 HR 30 Roman 1000 were of so dark a red as to appear almost black. Other leaves were placed in a weak infusion of raw meat and in human saliva, and the glands were much darkened in 25 Roman 1000, and after 40 Roman 1000 were so dark as almost to deserve to be called black. Even immersion for a whole day in distilled water occasionally induces some aggregation within the glands, so that they become of a darker tint. In all these cases, the glands are affected in exactly the same manner as those of Drosera milk, however, which acts so energetically on Drosera, seems rather less effective on Drosophilum, for the glands were only slightly darkened by an immersion of 1 HR 20 Roman 1000, but became decidedly darker after 3 HRS leaves, which had been left for 7 HRS in an infusion of raw meat or in saliva, were placed in the solution of carbonate of ammonia, and the glands now became greenish, whereas if they had been first placed in the carbonate, they would have become black. In this latter case, the ammonia probably combines with the acid of the secretion, and therefore does not act on the coloring matter, but when the glands are first subjected to an organic, page 339, fluid, either the acid is consumed in the work of digestion, or the cell walls are rendered more permeable, so that the undecomposed carbonate enters and acts on the coloring matter. If a particle of the dry carbonate is placed on a gland, the purple color is quickly discharged, owing probably to an excess of the salt. The gland, moreover, is killed. Turning now to the action of organic substances, the glands on which bits of raw meat were placed became dark-colored, and in 18 HRS, their contents were conspicuously aggregated. Several glands, with bits of albumin and fibrin, were darkened in between 2 HRS and 3 HRS, but in one case the purple color was completely discharged. Some glands which had caught flies were compared with others close by, and though they did not differ much in color, there was a marked difference in their state of aggregation. In some few instances, however, there was no such difference, and this appeared to be due to the insects having been caught long ago, so that the glands had recovered their pristine state. In one case, a group of the sessile colorless glands, to which a small fly adhered, presented a peculiar appearance, for they had become purple, owing to purple granular matter coating the cell walls. I may here mention, as a caution, that soon after some of my plants arrived in the spring from Portugal, the glands were not plainly acted on by bits of meat or insects, or a solution of ammonia, circumstance for which I cannot account. Digestion of solid animal matter. Whilst I was trying to place on 2 of the taller glands little cubes of albumin, these slipped down, and besmeared with secretion, were left resting on some of the small sessile glands. After 24 HRS, one of these cubes was found, page 340, completely liquefied, but with a few white streaks still visible. The other was much rounded, but not quite dissolved. 2 other cubes were left on tall glands for 2 HRS 45 Roman 1000, by which time all the secretion was absorbed, but they were not perceptibly acted on, though no doubt some slight amount of animal matter had been absorbed from them. They were then placed on the small sessile glands, which, being thus stimulated, secreted copiously in the course of 7 HRS. One of these cubes was much liquefied within this short time, and both were completely liquefied after 21 HRS 15 Roman 1000, the little liquid masses, however, still showing some white streaks. These streaks disappeared after an additional period of 6 HRS 30 Roman 1000, and by next morning Roman 1 E 48 HRS. From the time when the cubes were first placed on the glands, the liquefied matter was wholly absorbed. A cube of albumin was left on another tall gland, which first absorbed the secretion, and after 24 HRS poured forth a fresh supply. This cube, now surrounded by secretion, was left on the gland for an additional 24 HRS, but was very little, if at all, acted on. We may therefore conclude either that the secretion from the tall glands has little power of digestion, though strongly acid, or that the amount poured forth from a single gland is insufficient to dissolve a particle of albumin, which within the same time would have been dissolved by the secretion from several of the small sessile glands. Owing to the death of my last plant, I was unable to ascertain which of these alternatives is the true one. 4-minute shreds of pure fibrin were placed, each resting on 1, 2, or 3 of the taller glands, in the course of 2 HRS 30 Roman 1000. The secretion was all absorbed, and the shreds were left almost dry. They, page 341, were then pushed on to the sessile glands. 1 shred after 2 HRS 30 Roman 1000 seemed quite dissolved, but this may have been a mistake. A second, when examined after 17 HRS 25 Roman 1000, was liquefied, but the liquid, as seen under the microscope, still contained floating granules of fibrin. The other 2 shreds were completely liquefied after 21 HRS 30 Roman 1000, but in one of the drops, a very few granules could still be detected. These, however, were dissolved after an additional interval of 6 HRS 30 Roman 1000, and the surface of the leaf for some distance all round was covered with limpid fluid. It thus appears that Drosophilum digests albumin and fibrin rather more quickly than Drosera can, and this may perhaps be attributed to the acid, together probably with some small amount of the ferment being present in the secretion before the glands have been stimulated, so that digestion begins at once. Concluding remarks: the linear leaves of Drosophilum differ but slightly from those of certain species of Drosera, the chief differences being firstly the presence of minute almost sessile glands, which, like those of Dionea, do not secrete, until they are excited by the absorption of nitrogenous matter, but glands of this kind are present on the leaves of Drosera binata and appear to be represented by the papillae on the leaves of Drosera rotundifolia. Secondly, the presence of tentacles on the backs of the leaves, but we have seen that a few tentacles irregularly placed and tending towards abortion are retained on the backs of the leaves of Drosera binata. There are greater differences in function between the two genera. The most important one is that the tentacles of Drosophilum have no power of movement, this loss being partially replaced by the drops of viscid page 342 secretion being readily withdrawn from the glands, so that when an insect comes into contact with a drop, it is able to crawl away, but soon touches other drops, and then smothered by the secretion sinks down on the sessile glands and dies. Another difference is that the secretion from the tall glands, before they have been in any way excited, is strongly acid, and perhaps contains a small quantity of the proper ferment. Again, these glands do not secrete more copiously from being excited by the absorption of nitrogenous matter. On the contrary, they then absorb their own secretion with extraordinary quickness. In a short time, they begin to secrete again. All these circumstances are probably connected with the fact that insects do not commonly adhere to the glands with which they first come into contact, though this does sometimes occur, and that it is chiefly the secretion from the sessile glands which dissolves animal matter out of their bodies. Roridula dentata, this plant, a native of the western parts of the Cape of Good Hope, was sent to me in a dried state from Kew. It has an almost woody stem and branches, and apparently grows to a height of some feet. The leaves are linear with their summits, much attenuated. Their upper and lower surfaces are concave with a ridge in the middle, and both are covered with tentacles which differ greatly in length, some being very long, especially those on the tips of the leaves, and some very short. The glands also differ much in size and are somewhat elongated. They are supported on multicellular pedicels. This plant therefore agrees in several respects with page 343 Drosophilum, but differs in the following points. I could detect no sessile glands, nor would these have been of any use, as the upper surface of the leaves is thickly clothed with pointed unicellular hairs directed upwards. The pedicels of the tentacles do not include spiral vessels, nor are there any spiral cells within the glands. The leaves often arise in tufts and are pinnatophyid, the divisions projecting at right angles to the main linear blade. These lateral divisions are often very short and bear only a single terminal tentacle, with one or two short ones on the sides. No distinct line of demarcation can be drawn between the pedicels of the long terminal tentacles and the much attenuated summits of the leaves. We may indeed arbitrarily fix on the point to which the spiral vessels proceeding from the blade extend, but there is no other distinction. It was evident from the many particles of dirt sticking to the glands that they secrete much viscid matter. A large number of insects of many kinds also adhered to the leaves. I could nowhere discover any signs of the tentacles having been inflected over the captured insects, and this probably would have been seen even in the dried specimens had they possessed the power of movement. Hence, in this negative character, Roridula resembles its northern representative Drosophilum. Biblis gigantea, Western Australia, a dried specimen, about 18 inches in height with a strong stem, was sent me from Kew. The leaves are some inches in length, linear, slightly flattened, with a small projecting rib on the lower surface. They are covered on all sides by glands of two kinds, page 344, sessile ones arranged in rows, and others supported on moderately long pedicels. Towards the narrow summits of the leaves, the pedicels are longer than elsewhere, and here equal the diameter of the leaf. The glands are purplish, much flattened, and formed of a single layer of radiating cells, which in the larger glands are from 40 to 50. In number, the pedicels consist of single elongated cells with colorless, extremely delicate walls, marked with the finest intersecting spiral lines. Whether these lines are the result of contraction from the drying of the walls, I do not know, but the whole pedicel was often spirally rolled up. These glandular hairs are far more simple in structure than the so-called tentacles of the preceding genera, and they do not differ essentially from those borne by innumerable other plants. The flower peduncles bear similar glands. The most singular character about the leaves is that the apex is enlarged into a little knob covered with glands, and about a third broader than the adjoining part of the attenuated leaf. In two places, dead flies adhered to the glands, as no instance is known of unicellular structures, having any power of movement. Biblis no doubt catches insects solely by the aid of its viscid secretion. These probably sink down besmeared with the secretion and rest on the small sessile glands, which, if we may judge by the analogy of Drosophilum, then pour forth their secretion and afterwards absorb the digested matter. Supplementary observations on the power of absorption by the glandular hairs of other plants. A few observations on this subject may be here conveniently introduced, as the glands of many probably of all. Sachs Trait de Bot 3rd edit, 1874, p. 1026, page 345. The species of Droseraceae absorb fluids, or at least allow them readily to enter. It seemed desirable to ascertain how far the glands of other plants, which are not specially adapted for capturing insects, had the same power. Plants were chosen for trial at hazard, with the exception of 2 species of saxifrage, which were selected from belonging to a family allied to the Droseraceae. Most of the experiments were made by immersing the glands either in an infusion of raw meat or, more commonly, in a solution of carbonate of ammonia, as this latter substance acts so powerfully and rapidly on protoplasm. It seemed also particularly desirable to ascertain whether ammonia was absorbed, as a small amount is contained in rainwater. With the Droseraceae, the secretion of a viscid fluid by the glands does not prevent their absorbing, so that the glands of other plants might excrete superfluous matter or secrete an odoriferous fluid as a protection against the attacks of insects, or for any other purpose, and yet have the power of absorbing. I regret that in the following cases I did not try whether the secretion could digest or render soluble animal substances, but such experiments would have been difficult on account of the small size of the glands and the small amount of secretion. We shall see in the next chapter that the secretion from the glandular hairs of Pinguicula certainly dissolves animal matter. Saxifraga umbrosa, the flower peduncles and petioles of the leaves, are clothed with short hairs bearing pink-colored glands formed of several polygonal cells, with their pedicels divided by partitions into distinct cells, which are generally colorless, but sometimes pink. The glands secrete a yellowish viscid fluid. By the distinction between true absorption and mere permeation or imbibition is by no means clearly understood. See Muller's Physiology, Eng Translat, 1838, Vol. Roman, 1p. 280, page 346. Which minute diptera are sometimes, though not often, caught. The cells of the glands contain bright pink fluid charged with granules, or with globular masses of pinkish pulpy matter. This matter must be protoplasm, for it is seen to undergo slow but incessant changes of form. If a gland be placed in a drop of water and examined, similar movements were observed. After glands had been immersed in water for 13518 and 27 HRS, even after this latter period the glands retained their bright pink color, and the protoplasm within their cells did not appear to have become more aggregated. The continually changing forms of the little masses of protoplasm are not due to the absorption of water, as they were seen in glands kept dry. A flowerstem still attached to a plant was bent May 29 so as to remain immersed for 23 HRS 30 Roman 1000 in a strong infusion of raw meat. The color of the contents of the glands was slightly changed, being now of a duller and more purple tint than before. The contents also appeared more aggregated, for the spaces between the little masses of protoplasm were wider, but this latter result did not follow in some other and similar experiments. The masses seemed to change their forms more rapidly than did those in water, so that the cells had a different appearance. Every 4 or 5 minutes elongated masses became, in the course of 1 or 2 minutes, spherical, and spherical ones drew themselves out and united with others. Minute masses rapidly increased in size, and 3 distinct ones were seen to unite. The movements were, in short, exactly like those described in the case of Drosera. The cells of the pedicels were not affected by the infusion, nor were they in the following experiment. Another flowerstem was placed in the same manner, and for the same length of time, in a solution of 1 part of nitrate of ammonia to 146 of water, or 3 GRS to 1 Rs, and the glands were discolored in exactly the same manner as by the infusion of raw meat. Another flowerstem was immersed as before in a solution of 1 part of carbonate of ammonia to 109 of water. The glands, after 1 HR 30 Roman 1000, were not discolored, but after 3 HRS 45 Roman 1000, most of them had become dull purple, some of them blackish. In the case of Saxifraga tridactylites, Mr. Druce says, Pharmaceutical Journal, May 1875, that he examined some dozens of plants, and in almost every instance remnants of insects adhered to the leaves. So it is, as I hear from a friend, with this plant in Ireland, page 347. Green, a few being still unaffected, the little masses of protoplasm within the cells were seen in movement. The cells of the pedicels were unaltered. The experiment was repeated, and a fresh flowerstem was left for 23 HRS in the solution, and now a great effect was produced. All the glands were much blackened, and the previously transparent fluid in the cells of the pedicels, even down to their bases, contained spherical masses of granular matter. By comparing many different hairs, it was evident that the glands first absorbed the carbonate, and that the effect thus produced travels down the hairs from cell to cell. The first change which could be observed is a cloudy appearance in the fluid, due to the formation of very fine granules, which afterwards aggregate into larger masses altogether in the darkening of the glands, and in the process of aggregation traveling down the cells of the pedicels, there is the closest resemblance to what takes place when a tentacle of Drosera is immersed in a weak solution of the same salt. The glands, however, absorb very much more slowly than those of Drosera. Besides the glandular hairs, there are star-shaped organs which do not appear to secrete, and which were not in the least affected by the above solutions. Although in the case of uninjured flowerstems and leaves the carbonate seems to be absorbed only by the glands, yet it enters a cut surface much more quickly than a gland. Strips of the rind of a flowerstem were torn off, and the cells of the pedicels were seen to contain only colorless transparent fluid, those of the glands, including as usual some granular matter. These strips were then immersed in the same solution as before, 1 part of the carbonate to 109 of water, and in a few minutes granular matter appeared in the lower cells of all the pedicels. The action invariably commenced, for I tried the experiment repeatedly in the lowest cells, and therefore close to the torn surface, and then gradually traveled up the hairs until it reached the glands in a reverse direction to what occurs in uninjured specimens. The glands then became discolored, and the previously contained granular matter was aggregated into larger masses. 2 short bits of a flowerstem were also left for 2 HRS 40 Roman 1000 in a weaker solution of 1 part of the carbonate to 218 of water, and in both specimens the pedicels of the hairs near the cut ends now contained much granular matter, and the glands were completely discolored. Lastly, bits of meat were placed on some glands. These were examined after 23 HRS, as were others which had apparently not long before caught minute flies, but they did not present any page 348 difference from the glands of other hairs. Perhaps there may not have been time enough for absorption, I think. So as some glands on which dead flies had evidently long lain were of a pale dirty purple color, or even almost colorless, and the granular matter within them presented an unusual and somewhat peculiar appearance, that these glands had absorbed animal matter from the flies, probably by exosmose, into the viscid secretion. We may infer not only from their changed color, but because when placed in a solution of carbonate of ammonia, some of the cells in their pedicels become filled with granular matter, whereas the cells of other hairs, which had not caught flies, after being treated with the same solution for the same length of time, contained only a small quantity of granular matter. But more evidence is necessary before we fully admit that the glands of this saxifrage can absorb, even with ample time allowed, animal matter from the minute insects, which they occasionally and accidentally capture. Saxifraga rotundifolia, the hairs on the flowerstems of this species, are longer than those just described, and bear pale brown glands. Many were examined, and the cells of the pedicels were quite transparent. A bent stem was immersed for 30 Roman 1000 in a solution of 1 part of carbonate of ammonia to 109 of water, and 2 or 3 of the uppermost cells in the pedicels now contained granular or aggregated matter. The glands, having become of a bright yellowish green, the glands of this species therefore absorb the carbonate much more quickly than do those of Saxifraga umbrosa, and the upper cells of the pedicels are likewise affected much more quickly. Pieces of the stem were cut off and immersed in the same solution, and now the process of aggregation traveled up the hairs in a reverse direction, the cells close to the cut surfaces being first affected. Primula sinensis, the flower stems, the upper and lower surfaces of the leaves, and their footstalks are all clothed with a multitude of longer and shorter hairs. The pedicels of the longer hairs are divided by transverse partitions into 8 or 9 cells. The enlarged terminal cell is globular, forming a gland, which secretes a variable amount of thick, slightly viscid, not acid, brownish-yellow matter. A piece of a young flower stem was first immersed in distilled water for 2 HRS 30 Roman 1000, and the glandular hairs were not at all affected. Another piece, bearing 25 short and 9 long hairs, was carefully examined. The glands of the latter contained no solid or semi-solid matter, and those of only 2. Page 349 of the 25 short hairs contained some globules. This piece was then immersed for 2 HRS in a solution of 1 part of carbonate of ammonia to 109 of water, and now the glands of the 25 shorter hairs, with 2 or 3 exceptions, contained either 1 large or from 2 to 5 smaller spherical masses of semi-solid matter. 3 of the glands of the 9 long hairs likewise included similar masses. In a few hairs, there were also globules in the cells immediately beneath the glands. Looking to all 34 hairs, there could be no doubt that the glands had absorbed some of the carbonate. Another piece was left for only 1 HR in the same solution, and aggregated matter appeared in all the glands. My son Francis examined some glands of the longer hairs, which contained little masses of matter before they were immersed in any solution, and these masses slowly changed their forms, so that no doubt they consisted of protoplasm. He then irrigated these hairs for 1 HR 15 Roman 1000 whilst under the microscope, with a solution of 1 part of the carbonate to 218 of water. The glands were not perceptibly affected, nor could this have been expected, as their contents were already aggregated. But in the cells of the pedicels, numerous almost colorless spheres of matter appeared, which changed their forms and slowly coalesced, the appearance of the cells being thus totally changed at successive intervals of time. The glands on a young flower stem, after having been left for 2 HRS 45 Roman 1000 in a strong solution of 1 part of the carbonate to 109 of water, contained an abundance of aggregated masses, but whether generated by the action of the salt, I do not know. This piece was again placed in the solution so that it was immersed altogether for 6 HRS 15 Roman 1000, and now there was a great change, for almost all the spherical masses within the gland cells had disappeared, being replaced by granular matter of a darker brown. The experiment was thrice repeated with nearly the same result. On one occasion, the piece was left immersed for 8 HRS 30 Roman 1000, and though almost all the spherical masses were changed into the brown granular matter, a few still remained. If the spherical masses of aggregated matter had been originally produced merely by some chemical or physical action, it seems strange that a somewhat longer immersion in the same solution should so completely alter their character. But as the masses which slowly and spontaneously changed their forms must have consisted of living protoplasm, there is nothing surprising in its being injured or killed, and its appearance wholly changed by long immersion in so strong a solution of the carbonate, as that page 350 employed a solution of this strength. Paralyses all movement in Drosera, but does not kill the protoplasm. A still stronger solution prevents the protoplasm from aggregating into the ordinary full-sized globular masses, and these, though they do not disintegrate, become granular and opaque. In nearly the same manner, too, hot water and certain solutions, for instance of the salts of soda and potash, cause at first an imperfect kind of aggregation in the cells of Drosera, the little masses afterwards breaking up into granular or pulpy brown matter. All the foregoing experiments were made on flower stems, but a piece of a leaf was immersed for 30 Roman 1000 in a strong solution of the carbonate 1 part to 109 of water, and little globular masses of matter appeared in all the glands which before contained only limpid fluid. I made also several experiments on the action of the vapor of the carbonate on the glands, but will give only a few cases. The cut end of the footstalk of a young leaf was protected with sealing wax, and was then placed under a small bell glass with a large pinch of the carbonate. After 10 Roman 1000, the glands showed a considerable degree of aggregation, and the protoplasm lining the cells of the pedicels was a little separated from the walls. Another leaf was left for 50 Roman 1000, with the same result, excepting that the hairs became throughout their whole length of a brownish color. In a third leaf, which was exposed for 1 HR 50 Roman 1000, there was much aggregated matter in the glands, and some of the masses showed signs of breaking up into brown granular matter. This leaf was again placed in the vapor, so that it was exposed altogether for 5 HRS 30 Roman 1000. And now, though I examined a large number of glands, aggregated masses were found in only 2 or 3 in all the others. The masses which before had been globular were converted into brown opaque granular matter. We thus see that exposure to the vapor for a considerable time produces the same effects as long immersion in a strong solution. In both cases, there could hardly be a doubt that the salt had been absorbed chiefly or exclusively by the glands. On another occasion, bits of damp fibrin drops of a weak infusion of raw meat and of water were left for 24 HRS on some leaves. The hairs were then examined, but to my surprise differed in no respect from others, which had not been touched by these fluids. Most of the cells, however, included hyaline motionless little spheres, which did not seem to consist of protoplasm, but I suppose of some balsam or essential oil. Pelargonium zonilvar edged with white. The leaves, page 351, are clothed with numerous multicellular hairs, some simply pointed, others bearing glandular heads and differing much in length. The glands on a piece of leaf were examined and found to contain only limpid fluid. Most of the water was removed from beneath the covering glass, and a minute drop of 1 part of carbonate of ammonia to 146 of water was added, so that an extremely small dose was given. After an interval of only 3 Roman 1000, there were signs of aggregation within the glands of the shorter hairs, and after 5 Roman 1000, many small globules of a pale brown tint appeared in all of them. Similar globules, but larger, being found in the large glands of the longer hairs. After the specimen had been left for 1 HR in the solution, many of the smaller globules had changed their positions, and 2 or 3 vacuoles or small spheres, for I know not which they were, of a rather darker tint, appeared within some of the larger globules. Little globules could now be seen in some of the uppermost cells of the pedicels, and the protoplasmic lining was slightly separated from the walls of the lower cells. After 2 HRS 30 Roman 1000, from the time of first immersion, the large globules within the glands of the longer hairs were converted into masses of darker brown granular matter. Hence, from what we have seen with primula sinensis, there can be little doubt that these masses originally consisted of living protoplasm. A drop of a weak infusion of raw meat was placed on a leaf, and after 2 HRS 30 Roman 1000, many spheres could be seen within the glands. These spheres, when looked at again after 30 Roman 1000, had slightly changed their positions and forms, and one had separated into 2, but the changes were not quite like those which the protoplasm of Drosera undergoes. These hairs, moreover, had not been examined before immersion, and there were similar spheres in some glands which had not been touched by the infusion. Erica tetralix, a few long glandular hairs project from the margins of the upper surfaces of the leaves. The pedicels are formed of several rows of cells and support rather large globular heads secreting viscid matter, by which minute insects are. Occasionally, though rarely caught, some leaves were left for 23 HRS in a weak infusion of raw meat and in water, and the hairs were then compared, but they differed very little or not at all in both cases. The contents of the cells seemed rather more granular than they were before, but the granules did not exhibit any movement. Other leaves were left for 23 HRS in a solution of 1 part of carbonate of ammonia to 218 of water, and here again the granular matter appeared to have increased. Page 352 in amount, but one such mass retained exactly the same form as before. after an interval of 5 HRS, so that it could hardly have consisted of living protoplasm. These glands seem to have very little or no power of absorption, certainly much less than those of the foregoing plants. Mirabilis longiflora, the stems and both surfaces of the leaves, bear viscid hairs. Young plants, from 12 to 18 inches in height, in my greenhouse, caught so many minute Diptera coleoptera, and larvae that they were quite dusted with them. The hairs are short of unequal lengths, formed of a single row of cells, surmounted by an enlarged cell, which secretes viscid matter. These terminal cells or glands contain granules and often globules of granular matter within a gland which had caught a small insect. One such mass was observed to undergo incessant changes of form, with the occasional appearance of vacuoles. But I do not believe that this protoplasm had been generated by matter absorbed from the dead insect, for on comparing several glands which had and had not caught insects, not a shade of difference could be perceived between them, and they all contained fine granular matter. A piece of leaf was immersed for 24 HRS in a solution of 1 part of carbonate of ammonia to 218 of water, but the hairs seemed very little affected by it, excepting that perhaps the glands were rendered rather more opaque in the leaf itself. However, the grains of chlorophyll near the cut surfaces had run together or become aggregated, nor were the glands on another leaf after an immersion for 24 HRS in an infusion of raw meat in the least affected, but the protoplasm lining the cells of the pedicels had shrunk greatly from the walls. This latter effect may have been due to exosmose. As the infusion was strong, we may therefore conclude that the glands of this plant either have no power of absorption, or that the protoplasm which they contain is not acted on by a solution of carbonate of ammonia, and this seems scarcely credible, or by an infusion of meat. Nicotiana tabacum. This plant is covered with innumerable hairs of unequal lengths, which catch many minute insects. The pedicels of the hairs are divided by transverse partitions, and the secreting glands are formed of many cells containing greenish matter, with little globules of some substance. Leaves were left in an infusion of raw meat and in water for 26 HRS, but presented no difference. Some of these same leaves were then left for above 2 HRS in a solution of carbonate of ammonia, but no effect was produced. I regret that other experiments were not tried with more care, as Roman 1000 Schlossing, page 353, has shown that tobacco plants supplied with the vapor of carbonate of ammonia yield, on analysis, a greater amount of nitrogen than other plants not thus treated, and from what we have seen, it is probable that some of the vapor may be absorbed by the glandular hairs. A summary of the observations on glandular hairs. From the foregoing observations, few as they are, we see that the glands of 2 species of Saxifraga, of a primula and Pelargonium, have the power of rapid absorption, whereas the glands of an Erica mirabilis and Nicotiana either have no such power, or the contents of the cells are not affected by the fluids employed, namely a solution of carbonate of ammonia and an infusion of raw meat. As the glands of the mirabilis contain protoplasm, which did not become aggregated from exposure to the fluids just named, though the contents of the cells in the blade of the leaf were greatly affected by carbonate of ammonia, we may infer that they cannot absorb. We may further infer that the innumerable insects caught by this plant are of no more service to it than are those which adhere to the deciduous and sticky scales of the leaf buds of the horse chestnut. The most interesting case for us is that of the 2 species of Saxifraga, as this genus is distantly allied to Drosera. Their glands absorb matter from an infusion of raw meat, from solutions of the nitrate and carbonate of ammonia, and apparently from decayed insects. This was shown by the changed dull purple color of the protoplasm within the cells of the glands, by its state of aggregation, and apparently by its more rapid spontaneous movements. Comps Rendus, June 15, 1874. A good abstract of this paper is given in the Gardener's Chronicle, July 11, 1874, page 354. The aggregating process spreads from the glands down the pedicels of the hairs, and we may assume that any matter which is absorbed ultimately reaches the tissues of the plant. On the other hand, the process travels up the hairs whenever a surface is cut and exposed to a solution of the carbonate of ammonia. The glands on the flower stalks and leaves of Primula sinensis quickly absorb a solution of the carbonate of ammonia, and the protoplasm which they contain becomes aggregated. The process was seen in some cases to travel from the glands into the upper cells of the pedicels exposure for 10 Roman 1000 to the vapor of this salt likewise induced aggregation when leaves were left from 6 HRS to 7 HRS in a strong solution, or were long exposed to the vapor. The little masses of protoplasm became disintegrated brown and granular, and were apparently killed. An infusion of raw meat produced no effect on the glands. The limpid contents of the glands of Pelargonium zonale became cloudy and granular in from 3 Roman 1000 to 5 Roman 1000 when they were immersed in a weak solution of the carbonate of ammonia, and in the course of 1 HR granules appeared in the upper cells of the pedicels. As the aggregated masses slowly changed their forms, and as they suffered disintegration when left for a considerable time in a strong solution, there can be little doubt that they consisted of protoplasm. It is doubtful whether an infusion of raw meat produced any effect. The glandular hairs of ordinary plants have generally been considered by physiologists to serve only as secreting or excreting organs, but we now know that they have the power, at least in some cases, of absorbing both a solution and the vapor of ammonia, as rainwater contains a small percentage of ammonia, and the atmosphere a minute quantity of the carbonate. This page 355 power can hardly fail to be beneficial, nor can the benefit be quite so insignificant as it might at first be thought, for a moderately fine plant of Primula sinensis bears the astonishing number of above 2.5 millions of glandular hairs, all of which are able to absorb ammonia brought to them by the rain. It is moreover probable that the glands of some of the above-named plants obtain animal matter from the insects, which are occasionally entangled by the viscid secretion. Concluding remarks on the Drones race. The 6 known genera composing this family have now been described in relation to our present subject. As far as my means have permitted, they all capture insects. This is effected by Drosophilum, Rorigula, and Biblis solely by the viscid fluids secreted from their glands by Drosera through the same means, together with the movements of the tentacles by Dionea and Aldrovanda through the closing of the blades of the leaf. In these 2 last genera rapid. My son Francis counted the hairs on a space measured by means of a micrometer, and found that there were 35,336 on a square inch of the upper surface of a leaf, and 30,035 on the lower surface, that is, in about the proportion of 100 on the upper to 85 on the lower surface. On a square inch of both surfaces, there were 65,371 hairs. A moderately fine plant, bearing 12 leaves, the larger ones being a little more than 2 inches in diameter, was now selected, and the area of all the leaves together with their footstalks, the flower stems not being included, was found by a planimeter to be 39 square inches, so that the area of both surfaces was 78 square inches. Thus the plant, excluding the flower stems, must have borne the astonishing number of 2,568,099 glandular hairs. The hairs were counted late in the autumn, and by the following spring the leaves of some other plants of the same lot were found to be from 1/3 to 1/4 broader and longer than they were before, so that no doubt the glandular hairs had increased in number, and probably now much exceeded 3 millions. Page 356. Movement makes up for the loss of viscid secretion in every case. It is some part of the leaf which moves in Aldrovanda. It appears to be the basal parts alone which contract and carry with them the broad, thin margins of the lobes. In Dionea, the whole lobe, with the exception of the marginal prolongations or spikes, curves inwards, though the chief seat of movement is near the midrib. In Drosera, the chief seat is in the lower part of the tentacles, which homologically may be considered as prolongations of the leaf, but the whole blade often curls inwards, converting the leaf into a temporary stomach. There can hardly be a doubt that all the plants belonging to these 6 genera have the power of dissolving animal matter by the aid of their secretion, which contains an acid together with a ferment almost identical in nature with pepsin, and that they afterwards absorb the matter, thus digested. This is certainly the case with Drosera, Drosophilum, and Dionea, almost certainly with Aldrovanda, and from analogy very probable with Rorigula and Biblis. We can thus understand how it is that the 3 first-named genera are provided with such small roots, and that Aldrovanda is quite rootless. About the roots of the two other genera, nothing is known. It is no doubt a surprising fact that a whole group of plants, and as we shall presently see some other plants not allied to the Droseraceae, should subsist partly by digesting animal matter, and partly by decomposing carbonic acid, instead of exclusively, by this latter means together with the absorption of matter from the soil. By the aid of roots, we have however an equally anomalous case in the animal kingdom. The rhizocephalous crustaceans do not feed like other animals by their mouths, for they are destitute of an page 357 alimentary canal, but they live by absorbing through root-like processes the juices of the animals on which they are parasitic. Of the 6 genera, Drosera has been incomparably the most successful in the battle for life, and a large part of its success may be attributed to its manner of catching insects. It is a dominant form, for it is believed to include about 100 species, which range in the Old World from the Arctic regions to southern India, to the Cape of Good Hope, Madagascar and Australia, and in the New World from Canada to Tierra del Fuego. In this respect, it presents a marked contrast with the 5 other genera, which appear to be failing groups. Dionea includes only a single species, which is confined to one district in Carolina. The 3 varieties or closely allied species of Aldrovanda, like so many water plants, have a wide range from Central Europe to Bengal and Australia. Drosophilum includes only one species limited to Portugal and Morocco. Roridula and Biblis each have it. Fritz Muller, Facts for Darwin and Trans, 1869, p. 139. The rhizocephalous crustaceans are allied to the cirripeds. It is hardly possible to imagine a greater difference than that between an animal with prehensile limbs, a well-constructed mouth and alimentary canal, and one destitute of all these organs and feeding by absorption through branching root-like processes. If one rare cirripede, the Aenelasma squalicola, had become extinct, it would have been very difficult to conjecture how so enormous a change could have been gradually effected, but as Fritz Muller remarks, we have in Aenelasma an animal in an almost exactly intermediate condition, for it has root-like processes embedded in the skin of the shark, on which it is parasitic, and its prehensile cirri and mouth, as described in my monograph on the Lepididae Res Soc. 1851, p. 169, are in a most feeble and almost rudimentary condition. Dr. R. Kosman has given a very interesting discussion on this subject in his Suctoria and Lepididae 1873. See also Dr. Doen der Ursprung der Wirbeltiere 1875, p. 77. Bentham and Hooker genera, Plantarum Australia, is the metropolis of the genus, 41 species having been described from this country, as Prof. Oliver informs me, page 358. Here from Prof. Oliver, 2 species, the former confined to the western parts of the Cape of Good Hope, and the latter to Australia. It is a strange fact that Dionea, which is one of the most beautifully adapted plants in the vegetable kingdom, should apparently be on the highroad to extinction. This is all the more strange, as the organs of Dionea are more highly differentiated than those of Drosera. Its filaments serve exclusively as organs of touch, the lobes for capturing insects, and the glands when excited for secretion, as well as for absorption, whereas with Drosera, the glands serve all these purposes and secrete without being excited. By comparing the structure of the leaves, their degree of complication, and their rudimentary parts in the 6 genera, we are led to infer that their common parent form partook of the characters of Drosophilum, Roridula, and Biblis. The leaves of this ancient form were almost certainly linear, perhaps divided, and bore on their upper and lower surfaces glands, which had the power of secreting and absorbing. Some of these glands were mounted on pedicels, and others were almost sessile, the latter secreting only when stimulated by the absorption of nitrogenous matter in Biblis. The glands consist of a single layer of cells supported on a unicellular pedicel. In Roridula, they have a more complex structure and are supported on pedicels formed of several rows of cells. In Drosophilum, they further include spiral cells, and the pedicels include a bundle of spiral vessels. But in these 3 genera, these organs do not possess any power of movement, and there is no reason to doubt that they are of the nature of hairs or trichomes. Although in innumerable instances foliar organs move when excited, no case is known of a trichome having such page 359 power. We are thus led to inquire how the so-called tentacles of Drosera, which are manifestly of the same general nature as the glandular hairs of the above three genera, could have acquired the power of moving. Many botanists maintain that these tentacles consist of prolongations of the leaf because they include vascular tissue, but this can no longer be considered as a trustworthy distinction. The possession of the power of movement on excitement would have been safer evidence. But when we consider the vast number of the tentacles on both surfaces of the leaves of Drosophilum and on the upper surface of the leaves of Drosera, it seems scarcely possible that each tentacle could have aboriginally existed as a prolongation of the leaf Roridula, perhaps, shows us how we may reconcile these difficulties with respect to the homological nature of the tentacles. The lateral divisions of the leaves of this plant terminate in long tentacles, and these include spiral vessels, which extend for only a short distance up them, with no line of demarcation between what is plainly the prolongation of the leaf and the pedicel of a glandular hair. Therefore, there would be nothing anomalous or unusual in the basal parts of these tentacles, which correspond with the marginal ones of Drosera acquiring the power of movement, and we know that in Drosera it is only the lower part which becomes inflected. But in order to understand how in this latter genus not only the marginal, but all the inner tentacles have become capable of movement, we must further assume either that through the principle of correlated development this Sachs Trait de Botanique, 3rd edit, 1874, p. 1026. Dr. Warming sur la différence entre les trichomes Copenhague, 1873, p. 6. Extrait des Wissenschaftliche Metallurgie de la Soche d'Hist. Nat de Copenhague Nos. 1012, 1872, page 360. Power was transferred to the basal parts of the hairs, or that the surface of the leaf has been prolonged upwards at numerous points, so as to unite with the hairs, thus forming the bases of the inner tentacles. The above-named three genera, namely Drosophilum, Roridula, and Biblis, which appear to have retained a primordial condition, still bear glandular hairs on both surfaces of their leaves, but those on the lower surface have since disappeared in the more highly developed genera, with the partial exception of one species Drosera binata. The small sessile glands have also disappeared in some of the genera, being replaced in Roridula by hairs, and in most species of Drosera by absorbent papillae. Drosera binata, with its linear and bifurcating leaves, is in an intermediate condition. It still bears some sessile glands on both surfaces of the leaves, and on the lower surface a few irregularly placed tentacles, which are incapable of movement. A further slight change would convert the linear leaves of this latter species into the oblong leaves of Drosera anglica, and these might easily pass into orbicular ones with footstalks like those of Drosera rotundifolia. The footstalks of this latter species bear multicellular hairs, which we have good reason to believe represent aborted tentacles. The parent form of Dionea and Aldrovanda seems to have been closely allied to Drosera, and to have had rounded leaves supported on distinct footstalks, and furnished with tentacles all round the circumference, with other tentacles and sessile glands on the upper surface. I think so because the marginal spikes of Dionea apparently represent the extreme marginal tentacles of Drosera, the 6, sometimes 8 sensitive filaments on the upper surface, as well as the more numerous ones in Aldrovanda, representing the central page 361 tentacles of Drosera, with their glands aborted, but their sensitiveness retained. Under this point of view, we should bear in mind that the summits of the tentacles of Drosera, close beneath the glands, are sensitive. The three most remarkable characters possessed by the several members of the Droseraceae consist in the leaves of some having the power of movement, when excited, in their glands secreting a fluid which digests animal matter, and in their absorption of the digested matter. Can any light be thrown on the steps by which these remarkable powers were gradually acquired? As the walls of the cells are necessarily permeable to fluids in order to allow the glands to secrete it, is not surprising that they should readily allow fluids to pass inwards, and this inward passage would deserve to be called an act of absorption if the fluids combined with the contents of the glands judging from the evidence above, given the secreting glands of many other plants can absorb salts of ammonia, of which they must receive small quantities from the rain. This is the case with two species of Saxifraga, and the glands of one of them apparently absorb matter from captured insects, and certainly from an infusion of raw meat. There is therefore nothing anomalous in the Droseraceae having acquired the power of absorption in a much more highly developed degree. It is a far more remarkable problem how the members of this family and Pinguicula, and as Dr. Hooker has recently shown, Nepenthes, could all have acquired the power of secreting a fluid which dissolves or digests animal matter. The 6 genera of the Droseraceae very probably inherited this power from a common progenitor, but this cannot apply to page 362. Pinguicula or Nepenthes, for these plants, are not at all closely related to the Droseraceae. But the difficulty is not nearly so great as it at first appears. Firstly, the juices of many plants contain an acid, and apparently any acid serves for digestion. Secondly, as Dr. Hooker has remarked in relation to the present subject in his address at Belfast 1874, and as Sachs repeatedly insists, the embryos of some plants secrete a fluid which dissolves albuminous substances out of the endosperm, although the endosperm is not actually united with, only in contact with the embryo. All plants, moreover, have the power of dissolving albuminous or protid substances such as protoplasm, chlorophyll, gluten, allurone, and of carrying them from one part to other parts of their tissues. This must be effected by a solvent, probably consisting of a ferment together with an acid. Now, in the case of plants, which are able to absorb already soluble matter from captured insects, though not capable of true digestion, the solvent just referred to, which must be occasionally present in the glands, would be apt to exude from the glands together with the viscid secretion, inasmuch as endosmose is accompanied by exosmose. If such exudation did ever occur, the solvent would act on the animal matter contained within the captured insects, and this would be an act of true digestion, as it cannot be doubted that this process would be of high service to plants. Traité de botanique, 3rd edit, 1874, p. 844 c. Also for following facts, pp. 6476, 828, 831. Since this sentence was written, I have received a paper by Gorup Besanes, Bericht der Deutschen Chem-Gesellschaft, Berlin 1874, p. 1478, who with the aid of Dr. H. Will has actually made the discovery that the seeds of the vetch contain a ferment which, when extracted by glycerine, dissolves albuminous substances such as fibrin and converts them into true peptones. Page 363. Growing in very poor soil, it would tend to be perfected through natural selection. Therefore, any ordinary plant having viscid glands, which occasionally caught insects, might thus be converted under favorable circumstances into a species capable of true digestion. It ceases therefore to be any great mystery how several genera of plants, in no way closely related together, have independently acquired this same power. As there exist several plants, the glands of which cannot, as far as is known, digest animal matter, yet can absorb salts of ammonia and animal fluids, it is probable that this latter power forms the first stage towards that of digestion. It might, however, happen under certain conditions that a plant, after having acquired the power of digestion, should degenerate into one capable only of absorbing animal matter in solution or in a state of decay, or the final products of decay, namely the salts of ammonia. It would appear that this has actually occurred to a partial extent with the leaves of Aldrovanda, the outer parts of which possess absorbent organs, but no glands fitted for the secretion of any digestive fluid, these being confined to the inner parts. Little light can be thrown on the gradual acquirement of the third remarkable character possessed by the more highly developed genera of the Droseraceae, namely the power of movement. When excited, it should, however, be borne in mind that leaves and their homologues as well as flower peduncles have gained this power in innumerable instances independently of inheritance from any common parent form, for instance in tendril bearers and leaf climbers. Roman 1e plants with their leaves, petioles, and flower peduncles. Roman 100, modified for prehension, belonging to a large page 364 number of the most widely distinct order Sin, the leaves of the many plants which go to sleep at night or move, when shakened in the irritable stamens and pistils of not a few species, We may therefore infer that the power of movement can be, by some means, readily acquired. Such movements imply irritability or sensitiveness, but as Cohn has remarked, the tissues of the plants thus endowed do not differ in any uniform manner from those of ordinary plants. It is therefore probable that all leaves are to a slight degree irritable. Even if an insect alights on a leaf, a slight molecular change is probably transmitted to some distance across its tissue, with the sole difference that no perceptible effect is produced. We have some evidence in favor of this belief, for we know that a single touch on the glands of Drosera does not excite inflection, yet it must produce some effect, for if the glands have been immersed in a solution of camphor, inflection follows within a shorter time than would have followed from the effects of camphor alone. So again, with Dionea, the blades in their ordinary state may be roughly touched without their closing, yet some effect must be thus caused and transmitted across the whole leaf. For if the glands have recently absorbed animal matter, even a delicate touch causes them to close instantly. On the whole, we may conclude that the acquirement of a high degree of sensitiveness and of the power of movement by certain genera of the Droseraceae presents no greater difficulty than that presented by the similar but feebler powers of a multitude of other plants. See the abstract of his memoir on the contractile tissues of plants in the Annals and Mag of Nat. Hist. 3rd Series, Vol. Roman, 11 p. 188, page 365. The specialized nature of the sensitiveness possessed by Drosera and Dionea, and by certain other plants, well deserves attention. A gland of Drosera may be forcibly hit once, twice, or even thrice, without any effect being produced, whilst the continued pressure of an extremely minute particle excites movement. On the other hand, a particle many times heavier may be gently laid on one of the filaments of Dionea with no effect, but if touched only once by the slow movement of a delicate hair, the lobes close, and this difference in the nature of the sensitiveness of these two plants stands in manifest adaptation to their manner of capturing insects. So does the fact that when the central glands of Drosera absorb nitrogenous matter, they transmit a motor impulse to the exterior tentacles much more quickly than when they are mechanically irritated. Whilst with Dionea, the absorption of nitrogenous matter causes the lobes to press together with extreme slowness, whilst a touch excites rapid movement. Somewhat analogous cases may be observed, as I have shown in another work, with the tendrils of various plants, some being most excited by contact with fine fibers, others by contact with bristles, others with a flat or a creviced surface. The sensitive organs of Drosera and Dionea are also specialized so as not to be uselessly affected by the weight or impact of drops of rain or by blasts of air. This may be accounted for by supposing that these plants and their progenitors have grown accustomed to the repeated action of rain and wind, so that no molecular change is thus induced, whilst they have been rendered more sensitive by means of natural selection to the rarer impact or pressure of solid bodies. Although the absorption by the glands of Drosera of various fluids excites move page 366 meant, there is a great difference in the action of allied fluids, for instance, between certain vegetable acids, and between citrate and phosphate of ammonia. The specialized nature and perfection of the sensitiveness in these two plants is all the more astonishing as no one supposes that they possess nerves, and by testing Drosera with several substances which act powerfully on the nervous system of animals, it does not appear that they include any diffused matter analogous to nerve tissue. Although the cells of Drosera and Dionea are quite as sensitive to certain stimulants as are the tissues which surround the terminations of the nerves in the higher animals, yet these plants are inferior even to animals low down in the scale, in not being affected except by stimulants in contact with their sensitive parts. They would, however, probably be affected by radiant heat, for warm water excites energetic movement. When a gland of Drosera or one of the filaments of Dionea is excited, the motor impulse radiates in all directions, and is not, as in the case of animals, directed towards special points or organs. This holds good even in the case of Drosera, when some exciting substance has been placed at 2 points on the disc, and when the tentacles all round are inflected with marvelous precision towards the 2 points. The rate at which the motor impulse is transmitted, though rapid in Dionea, is much slower than in most or all animals. This fact, as well as that of the motor impulse not being specially directed to certain points, are both no doubt due to the absence of nerves. Nevertheless, we perhaps see the prefigurement of the formation of nerves in animals, in the transmission of the motor impulse being so much more rapid down the confined space within the tentacles of Drosera than page 367 elsewhere, and somewhat more rapid in a longitudinal than in a transverse direction across the disc. These plants exhibit still more plainly their inferiority to animals in the absence of any reflex action, except in so far as the glands of Drosera, when excited from a distance, send back some influence, which causes the contents of the cells to become aggregated down to the bases of the tentacles. But the greatest inferiority of all is the absence of a central organ able to receive impressions from all points, to transmit their effects in any definite direction, to store them up and reproduce them. Page 368. Chapter Romans 16. Pinguicula. Pinguicula vulgaris, structure of leaves, number of insects and other objects caught, movement of the margins of the leaves, uses of this movement, secretion, digestion, and absorption, action of the secretion on various animal and vegetable substances, the effects of substances not containing soluble nitrogenous matter on the glands. Pinguicula grandiflora, Pinguicula lusitanica, catches insects' movement of the leaves, secretion, and digestion. Pinguicula vulgaris, this plant grows in moist places, generally on mountains. It bears on an average 8 rather thick oblong light green leaves, having scarcely any footstock. A full-sized leaf is about 1/12 inch in length and 34 inch in breadth. The young central leaves are deeply concave and project upwards. The older ones towards the outside are flat or convex and lie close to the ground, forming a rosette. From 3 to 4 inches in diameter, the margins of the leaves are incurved. Their upper surfaces are thickly covered with 2 sets of glandular hairs differing in the size of the glands, and in the length of their pedicels. The larger glands have a circular outline as seen from above, and are of moderate thickness. They are divided by radiating partitions into 16 cells containing light green homogeneous fluid. They are supported on elongated unicellular pedicels containing a nucleus with a nucleolus which rest on slight prominences. The small glands differ only in being formed of about half the number of cells, containing much paler fluid, and supported on much shorter pedicels near the midrib towards the base of the leaf. The page 369 pedicels are multicellular, are longer than elsewhere, and bear smaller glands. All the glands secrete a colorless fluid, which is so viscid that I have seen a fine thread drawn out to a length of 18 inches. But the fluid in this case was secreted by a gland which had been excited. The edge of the leaf is translucent and does not bear any glands, and here the spiral vessels proceeding from the midrib terminate in cells marked by a spiral line, somewhat like those within the glands of Drosera. The roots are short. 3 plants were dug up in North Wales on June 20 and carefully washed. Each bore 5 or 6 unbranched roots, the longest of which was only 1 inch. 2 rather young plants were examined on September 28. These had a greater number of roots, namely 8 and 18, all under 1 inch in length, and very little branched. I was led to investigate the habits of this plant by being told by Mr. W. Marshall that on the mountains of Cumberland many insects adhere to the leaves. A friend sent me on June 23 39 leaves from North Wales, which were selected owing to objects of some kind adhering to them. Of these leaves, 32 had caught 142 insects, or on an average 4 per leaf minute. Fragments of insects not being included. Besides the insects, small leaves belonging to 4 different kinds of plants, those of Erica tetralix being much the commonest, and 3-minute seedling plants blown by the wind adhered to 19 of the leaves. One had caught as many as 10 leaves of the Erica seeds or fruits, commonly of Kerx, and one of Juncus, besides bits of moss and other rubbish. Likewise adhered to 6 of the 39 leaves. The same friend on June 27 collected 9 plants bearing 7 to 4 leaves, and all of these, with the exception of 3 young leaves, had caught insects. 30 insects were counted on 1 leaf, 18 on a second, and 16 on a third. Another friend examined on August 22 some plants in Donegal, Ireland, and found insects on 70 out of 157 leaves 15 of page 370. These leaves were sent me each, having caught on an average 2 insects. To 9 of them, leaves mostly of Erica tetralix adhered, but they had been specially selected on this latter account. I may add that early in August my son found leaves of this same Erica and the fruits of a kerx on the leaves of a pinguicula in Switzerland, probably Pinguicula alpina. Some insects, but no great number, also adhered to the leaves of this plant, which had much better developed roots than those of Pinguicula vulgaris in Cumberland. Mr. Marshall on September 3 carefully examined for me 10 plants bearing 80 leaves, and on 63 of these Roman 1e, on 79%. He found insects 143 in number, so that each leaf had on an average 2 insects. A few days later, he sent me some plants with 16 seeds or fruits. Adhering to 14 leaves, there was a seed. On 3 leaves on the same plant, the 16 seeds belonged to 9 different kinds, which could not be recognized, excepting one of ranunculus and several belonging to 3 or 4 distinct species of kerx. It appears that fewer insects are caught late in the year than earlier. Thus in Cumberland, from 20 to 24 insects were observed in the middle of July on several leaves, whereas in the beginning of September the average number was only 2. Most of the insects in all the foregoing cases were diptera, but with many minute hymenoptera, including some ants, a few small coleoptera larvae, spiders, and even small moths. We thus see that numerous insects and other objects are caught by the viscid leaves, but we have no right to infer from this fact that the habit is beneficial to the plant, any more than in the before given case of the mirabilis or of the horsechestnut. But it will presently be seen that dead insects and other nitrogenous bodies excite the glands to increased secretion, and that the secretion then becomes acid and has the power of digesting animal substances such as albumin, fibrin, Roman 100. Moreover, the dissolved nitrogenous matter is absorbed by the glands, as shown by their limpid contents being aggregated into slowly moving granular masses of protoplasm. The same results follow when insects are naturally captured, and as the plant lives in poor soil and has small roots, there can be no page 371 doubt that it profits by its power of digesting and absorbing matter from the prey which it habitually captures in such large numbers. It will, however, be convenient first to describe the movements of the leaves. Movements of the leaves. That such thick, large leaves as those of Pinguicula vulgaris should have the power of curving inwards when excited has never even been suspected. It is necessary to select for experiment leaves with their glands secreting freely and which have been prevented from capturing. Many insects, as old leaves, at least those growing in a state of nature, have their margins already curled so much inwards that they exhibit little power of movement or move very slowly. I will first give in detail the more important experiments which were tried, and then make some concluding remarks. Experiment 1A. Young and almost upright leaf was selected with its 2 lateral edges equally and very slightly incurved. A row of small flies was placed along one margin. When looked at next day after 15 HRS, this margin, but not the other, was found folded inwards like the helix of the human ear to the breadth of 110 of an inch, so as to lie partly over the row of flies. Fig. 15. The glands on which the flies rested, as well as those on the overlapping margin, which had been brought into contact with the flies, were all secreting copiously. Fig. 15. Pinguicula vulgaris, outline of leaf, with left margin inflected over a row of small flies. Experiment 2. A row of flies was placed on one margin of a rather old leaf, which lay flat on the ground, and in this case, the margin after the same interval as before, namely 15 HRS, had only just begun to curl inwards, but so much secretion had been poured forth that the spoon-shaped tip of the leaf was filled with it. Experiment 3. Fragments of a large fly were placed close to the apex of a vigorous leaf, as well as along half one margin, page 372. After 4 HRS 20 Roman 1000, there was decided incurvation, which increased a little during the afternoon, but was in the same state on the following morning. Near the apex, both margins were inwardly curved. I have never seen a case of the apex itself being in the least curved towards the base of the leaf, after 48 HRS, always reckoning from the time when the flies were placed on the leaf. The margin had everywhere begun to unfold. Experiment 4. A large fragment of a fly was placed on a leaf in a medial line, a little beneath the apex. Both lateral margins were perceptibly incurved in 3 HRS, and after 4 HRS 20 Roman 1000 to such a degree that the fragment was clasped by both margins. After 24 HRS, the two enfolded edges near the apex for the lower part of the leaf was not at all affected, were measured, and found to be 11 of an inch 2 Roman 2000 apart. The fly was now removed, and a stream of water poured over the leaf so as to wash the surface, and after 24 HRS, the margins were 25 of an inch 6 Roman 2000 apart, so that they were largely unfolded. After an additional 24 HRS, they were completely unfolded. Another fly was now put on the same spot to see whether this leaf, on which the first fly had been left 24 HRS, would move again. After 10 HRS, there was a trace of incurvation, but this did not increase during the next 24 HRS. A bit of meat was also placed on the margin of a leaf which 4 days previously had become strongly incurved over a fragment of a fly and had afterwards re-expanded, but the meat did not cause even a trace of incurvation. On the contrary, the margin became somewhat reflexed, as if injured, and so remained for the 3 following days, as long as it was observed. Experiment 5. A large fragment of a fly was placed halfway between the apex and base of a leaf, and halfway between the midrib and one margin. A short space of this margin opposite, the fly showed a trace of incurvation after 3 HRS, and this became strongly pronounced in 7 HRS. After 24 HRS, the enfolded edge was only 16 of an inch 4 Roman 2000 from the midrib. The margin now began to unfold, though the fly was left on the leaf, so that by the next morning Roman 1 E48 HRS, from the time when the fly was first put on the enfolded edge, had almost completely recovered its original position, being now 3 of an inch 7 Roman 2000, instead of 16 of an inch, from the midrib. A trace of flexure was, however, still visible. Experiment 6. A young and concave leaf was selected with its margins slightly and naturally incurved. 2 rather large oblong rectangular pieces of roast meat were placed with their ends touching the enfolded edge, and 46 of an inch 11 Roman 2000 page 373 apart from one another. After 24 HRS, the margin was greatly and equally incurved, see Fig 16, throughout this space, and for a length of 12 or 13 of an inch 3 or 3 Roman 2000 above and below each bit, so that the margin had been affected over a greater length between the 2 bits owing to their conjoint action than beyond them. The bits of meat were too large to be clasped by the margin, but they were tilted up one of them so as to stand almost vertically. After 48 HRS, the margin was almost unfolded, and the bits had sunk down. When again examined after 2 days, the margin was quite unfolded, with the exception of the naturally inflected edge, and one of the bits of meat, the end of which had at first touched the edge, was now 67 of an inch 1 Roman 2000 distant from it, so that this bit had been pushed thus far across the blade of the leaf. Fig 16, Pinguicula vulgaris, outline of leaf, with right margin inflected against 2 square bits of meat. Experiment 7. A bit of meat was placed close to the incurved edge of a rather young leaf, and after it had re-expanded, the bit was left lying 11 of an inch 2 Roman 2000 from the edge. The distance from the edge to the midrib of the fully expanded leaf was 35 of an inch 8 Roman 2000, so that the bit had been pushed inwards and across nearly 1/3 of its semi-diameter. Experiment 8. Cubes of sponge soaked in a strong infusion of raw meat were placed in close contact with the incurved edges of 2 leaves. An older and younger one. The distance from the edges to the midribs was carefully measured. After 1 HR 17 Roman 1000, there appeared to be a trace of incurvation. After 2 HRS 17 Roman 1000, both leaves were plainly inflected, the distance between the edges and midribs being now only half what it was at first. The incurvation increased slightly during the next 412 HRS, but remained nearly the same for the next 17 HRS 30 Roman 1000 in 35 HRS. From the time when the sponges were placed on the leaves, the margins were a little unfolded to a greater degree in the younger than in the older leaf. The latter was not quite unfolded until the third day, and now both bits of sponge were left at the distance of 1/2 Roman 2000 from the edges, or about a quarter of the distance between the edge and midrib. A third bit of sponge adhered to the edge, and as the margin unfolded, was dragged backwards into its original position, page 374. Experiment 9. A chain of fibers of roast meat as thin as bristles and moistened with saliva were placed down one whole side close to the narrow, naturally incurved edge of a leaf in 3 HRS. This side was greatly incurved along its whole length, and after 8 HRS formed a cylinder about 120 of an inch 1 diameter, quite concealing the meat. This cylinder remained closed for 32 HRS, but after 48 HRS was half unfolded, and in 72 HRS was as open as the opposite margin, where no meat had been placed. As the thin fibers of meat were completely overlapped by the margin, they were not pushed at all inwards across the blade. Experiment 10.6. Cabbage seeds soaked for a night in water were placed in a row close to the narrow incurved edge of a leaf. We shall hereafter see that these seeds yield soluble matter to the glands. In 2 HRS 25 Roman 1000 the margin was decidedly inflected. In 4 HRS it extended over the seeds for about half their breadth, and in 7 HRS over 3/4 of their breadth, forming a cylinder not quite closed along the inner side, and about 7 of an inch 1 Roman 2000 in diameter. After 24 HRS, the inflection had not increased, perhaps had decreased. The glands, which had been brought into contact with the upper surfaces of the seeds, were now secreting freely. In 36 HRS, from the time when the seeds were put on the leaf, the margin had greatly, and after 48 HRS had completely re-expanded, as the seeds were no longer held by the inflected margin, and as the secretion was beginning to fail, they rolled some way down the marginal channel. Experiment 11. Fragments of glass were placed on the margins of 2 fine young leaves. After 2 HRS 30 Roman 1000, the margin of one certainly became slightly incurved, but the inflection never increased and disappeared. In 16 HRS 30 Roman 1000, from the time when the fragments were first applied. With the second leaf, there was a trace of incurvation in 2 HRS 15 Roman 1000, which became decided in 4 HRS 30 Roman 1000, and still more strongly pronounced in 7 HRS. But after 19 HRS 30 Roman 1000 had plainly decreased, the fragments excited at most a slight and doubtful increase of the secretion, and in 2 other trials no increase could be perceived. Bits of coal cinders placed on a leaf produced no effect, either owing to their lightness or to the leaf being torpid. Experiment 12. We now turn to fluids. A row of drops of a strong infusion of raw meat were placed along the margins of 2 leaves, squares of sponge soaked in the same infusion being placed on the opposite margins. My object was to assert, page 375, whether a fluid would act as energetically as a substance, yielding the same soluble matter to the glands. No distinct difference was perceptible, certainly none in the degree of incurvation, but the incurvation round the bits of sponge lasted rather longer, as might perhaps have been expected from the sponge remaining damp, and supplying nitrogenous matter for a longer time. The margins with the drops became plainly incurved. In 2 HRS 17 Roman 1000, the incurvation subsequently increased somewhat, but after 24 HRS had greatly decreased. Experiment 13. Drops of the same strong infusion of raw meat were placed along the midrib of a young and rather deeply concave leaf. The distance across the broadest part of the leaf between the naturally incurved edges was 55 of an inch 13 Roman 2000 in 3 HRS 27 Roman 1000. This distance was a trace less in 6 HRS 27 Roman 1000. It was exactly 45 of an inch 11 Roman 2000, and had therefore decreased by 1 of an inch 2 Roman 2000. After only 10 HRS 37 Roman 1000, the margin began to re-expand, for the distance from edge to edge was now a trace wider, and after 24 HRS 20 Roman 1000 was as great within a hair's breadth as when the drops were first placed on the leaf. From this experiment, we learn that the motor impulse can be transmitted to a distance of 22 of an inch 5 Roman 2000 in a transverse direction from the midrib to both margins, but it would be safer to say 2 of an inch 5 Roman 2000, as the drops spread a little beyond the midrib. The incurvation thus caused lasted for an unusually short time. Experiment 14. 3 drops of a solution of 1 part of carbonate of ammonia to 218 of water 2 grs to 1 Rs were placed on the margin of a leaf. These excited so much secretion that in 1H22 Roman 1000 all 3 drops ran together, but although the leaf was observed for 24 HRS, there was no trace of inflection. We know that a rather strong solution of this salt, though it does not injure the leaves of Drosera, paralyses their power of movement, and I have no doubt from the following case that this holds good with Pinguicula. Experiment 15. A row of drops of a solution of 1 part of carbonate of ammonia to 875 of water 1 gr to 2 Rs was placed on the margin of a leaf in 1 HR. There was apparently some slight incurvation, and this was well marked in 3 HRS 30 Roman 1000. After 24 HRS, the margin was almost completely re-expanded. Experiment 16. A row of large drops of a solution of 1 part of phosphate of ammonia to 4375 of water 1 gr to 10 Rs was placed along the margin of a leaf. No effect was produced, and after 8 HRS fresh drops were added along the same margin without the least effect. We know that a solution of this page 376 strength acts powerfully on Drosera, and it is just possible that the solution was too strong. I regret that I did not try a weaker solution. Experiment 17. As the pressure from bits of glass causes incurvation, I scratched the margins of 2 leaves for some minutes with a blunt needle, but no effect was produced. The surface of a leaf, beneath a drop of a strong infusion of raw meat, was also rubbed for 10 Roman 1000 with the end of a bristle, so as to imitate the struggles of a captured insect, but this part of the margin did not bend sooner than the other parts, with undisturbed drops of the infusion. We learn from the foregoing experiments that the margins of the leaves curl inwards when excited by the mere pressure of objects not yielding any soluble matter, by objects yielding such matter, and by some fluids, namely an infusion of raw meat and a weak solution of carbonate of ammonia, a stronger solution of 2 grains of this salt to an ounce of water, though exciting copious secretion, paralyses the leaf. Drops of water and of a solution of sugar or gum did not cause any movement. Scratching the surface of the leaf for some minutes produced no effect. Therefore, as far as we at present know, only 2 causes, namely slight continued pressure and the absorption of nitrogenous matter, excite movement. It is only the margins of the leaf which bend, for the apex never curves towards the base. The pedicels of the glandular hairs have no power of movement. I observed on several occasions that the surface of the leaf became slightly concave where bits of meat or large flies had long lain, but this may have been due to injury from overstimulation. The shortest time in which plainly marked movement was observed was 2 HRS 17 Roman 1000, and this occurred when either nitrogenous substances or fluids were placed on the leaves, but I believe that in some cases, page 377, there was a trace of movement in 1 HR or 1 HR 30 Roman 1000. The pressure from fragments of glass excites movement almost as quickly as the absorption of nitrogenous matter, but the degree of incurvation thus caused is much less after a leaf has become well incurved and has again expanded. It will not soon answer to a fresh stimulus. The margin was affected longitudinally upwards or downwards for a distance of 13 of an inch 3 Roman 2000 from an excited point, but for a distance of 46 of an inch between 2 excited points, and transversely for a distance of 2 of an inch 5 Roman 2000. The motor impulse is not accompanied, as in the case of Drosera, by any influence causing increased secretion, for when a single gland was strongly stimulated and secreted copiously, the surrounding glands were not in the least affected. The incurvation of the margin is independent of increased secretion, for fragments of glass cause little or no secretion, and yet excite movement, whereas a strong solution of carbonate of ammonia quickly excites copious secretion, but no movement. One of the most curious facts with respect to the movement of the leaves is the short time during which they remain incurved. Although the exciting object is left on them, in the majority of cases, there was well-marked re-expansion within 24 HRS from the time when even large pieces of meat, Roman 100, were placed on the leaves, and in all cases within 48 HRS. In one instance, the margin of a leaf remained for 32 HRS, closely inflected round thin fibers of meat. In another instance, when a bit of sponge soaked in a strong infusion of raw meat had been applied to a leaf, the margin began to unfold in 35 HRS fragments of glass, keep the margin incurved for a shorter time than do nitrogenous bodies, for in the former case, there was page 378 complete re-expansion in 16 HRS, 30 Roman 1000 nitrogenous fluids act for a shorter time than nitrogenous substances. Thus, when drops of an infusion of raw meat were placed on the midrib of a leaf, the incurved margins began to unfold in only 10 HRS, 37 Roman 1000. And this was the quickest act of re-expansion observed by me, but it may have been partly due to the distance of the margins from the midrib where the drops lay. We are naturally led to inquire what is the use of this movement, which lasts for so short a time, if very small objects, such as fibers of meat, or moderately small objects, such as little flies or cabbage seeds, are placed close to the margin. They are either completely or partially embraced by it. The glands of the overlapping margin are thus brought into contact with such objects and pour forth their secretion, afterwards absorbing the digested matter. But as the incurvation lasts for so short a time, any such benefit can be of only slight importance, yet perhaps greater than at first appears. The plant lives in humid districts, and the insects which adhere to all parts of the leaf are washed by every heavy shower of rain into the narrow channel formed by the naturally incurved edges. For instance, my friend in North Wales placed several insects on some leaves, and 2 days afterwards, there having been heavy rain in the interval, found some of them quite washed away, and many others safely tucked under the now closely inflected margins, the glands of which all round the insects were no doubt secreting. We can thus also understand how it is that so many insects and fragments of insects are generally found lying within the incurved margins of the leaves. The incurvation of the margin due to the presence of an exciting object must be serviceable in another page 379, and probably more important way. We have seen that when large bits of meat or of sponge soaked in the juice of meat were placed on a leaf, the margin was not able to embrace them, but as it became incurved, pushed them very slowly towards the middle of the leaf, to a distance from the outside of fully 1 inch 2 Roman 2000, that is across. Between 1/3 and 1/4 of the space, between the edge and midrib, any object, such as a moderately sized insect, would thus be brought slowly into contact with a far larger number of glands, inducing much more secretion and absorption than would otherwise have been the case. That this would be highly serviceable to the plant, we may infer from the fact that Drosera has acquired highly developed powers of movement merely for the sake of bringing all its glands into contact with captured insects. So again, after a leaf of Dionea has caught an insect, the slow pressing together of the two lobes serves merely to bring the glands on both sides into contact with it, causing also the secretion charged with animal matter to spread by capillary attraction over the whole surface. In the case of Pinguicula, as soon as an insect has been pushed for some little distance towards the midrib, immediate re-expansion would be beneficial, as the margins could not capture fresh prey until they were unfolded. The service rendered by this pushing action, as well as that from the marginal glands being brought into contact for a short time with the upper surfaces of minute-captured insects, may perhaps account for the peculiar movements of the leaves. Otherwise, we must look at these movements as a remnant of a more highly developed power formerly possessed by the progenitors of the genus. In the 4 British species, and as I hear from page 380, Prof. Dyer, in most or all the species of the genus, the edges of the leaves are in some degree naturally and permanently incurved. This incurvation serves, as already shown, to prevent insects from being washed away by the rain, but it likewise serves for another end. When a number of glands have been powerfully excited by bits of meat, insects, or any other stimulus, the secretion often trickles down the leaf and is caught by the incurved edges instead of rolling off and being lost. As it runs down the channel, fresh glands are able to absorb the animal matter held. In solution, moreover, the secretion often collects in little pools within the channel or in the spoon-like tips of the leaves, and I ascertained that bits of albumin, fibrin, and gluten are here dissolved more quickly and completely than on the surface of the leaf, where the secretion cannot accumulate, and so it would be with naturally caught insects. The secretion was repeatedly seen thus to collect on the leaves of plants protected from the rain, and with exposed plants, there would be still greater need of some provision to prevent as far as possible the secretion with its dissolved animal matter being wholly lost. It has already been remarked that plants growing in a state of nature have the margins of their leaves much more strongly incurved than those grown in pots, and prevented from catching many insects. We have seen that insects washed down by the rain from all parts of the leaf often lodge within the margins, which are thus excited to curl farther inwards, and we may suspect that this action, many times repeated during the life of the plant, leads to their permanent and well-marked incurvation. I regret that this view did not occur to me in time to test its truth. It may here be added, though not immediately, page 381, bearing on our subject, that when a plant is pulled up, the leaves immediately curl downwards, so as almost to conceal the roots, a fact which has been noticed by many persons. I suppose that this is due to the same tendency which causes the outer and older leaves to lie flat on the ground. It further appears that the flower stalks are to a certain extent irritable, for Dr. Johnson states that they bend backwards if rudely handled. Secretion, absorption, and digestion. I will first give my observations and experiments, and then a summary of the results. The effects of objects containing soluble nitrogenous matter. English Botany by Sir J. E. Smith, with colored figures by J. Sowerby, edit of 1832, tab 24, 25, 26, page 382. 2 or 3 days, much reduced in size, rounded, rendered more or less colorless and transparent, and so much softened that they fell to pieces on the slightest touch. In only one instance was a very minute particle completely dissolved, and this occurred within 48 HRS, when only a small amount of secretion was excited. This was generally absorbed in from 24 HRS to 48 HRS, the glands being left dry. But when the supply of secretion was copious, round, either a single rather large bit of meat or round several small bits, the glands did not become dry until 6 or 7 days had elapsed. The most rapid case of absorption observed by me was when a small drop of an infusion of raw meat was placed on a leaf for the glands. Here became almost dry in 3 HRS 20 Roman 1000 glands, excited by small particles of meat, and which have quickly absorbed their own secretion, begin to secrete again in the course of 7 or 8 days from the time when the meat was given them. The 9 following kinds of seeds excited only a slight amount of secretion, namely celery, parsnip, caraway, linum grandiflorum, cassia, trifolium, panonicum, plantago, onion, page 386, and bromus. Most of these seeds did not excite any secretion until 48 HRS had elapsed, and in the case of the trifolium, only 1 seed acted, and this not until the 3rd day. Although the seeds of the plantago excited very little secretion, the glands continued to secrete for 6 days. Lastly, the 5 following kinds excited no secretion, though left on the leaves for 2 or 3 days, namely lettuce, erica, tetralix, atriplex, hortensis, phalaris canariensis, and wheat. Nevertheless, when the seeds of the lettuce, wheat, and atriplex were split open, and applied to leaves, secretion was excited in considerable quantity in 10 HRS, and I believe that some was excited in 6 hours in the case of the atriplex. The secretion ran down to the margin, and after 24 HRS, I speak of it in my notes as immense in quantity, and acid. The split seeds, also of the trifolium and celery, acted powerfully and quickly, though the whole seeds caused, as we have seen, very little secretion, and only after a long interval of time, a slice of the common pea, which however was not tried whole, caused secretion in 2 HRS. From these facts, we may conclude that the great difference in the degree and rate at which various kinds of seeds excite secretion is chiefly or wholly due to the different permeability of their coats. Some thin slices of the common pea which had been previously soaked for 1 hr in water, were placed on a leaf and quickly excited much acid secretion. After 24 hrs, these slices were compared under a high power with others left in water for the same time. The latter contained so many fine granules of legumin that the slide was rendered muddy, whereas the slices which had been subjected to the secretion were much cleaner and more transparent, the granules of legumin apparently having been dissolved a cabbage seed which had lain for 2 days on a leaf and had excited much acid secretion was cut into slices, and these were compared with those of a seed which had been left for the same time in water. Those subjected to the secretion were of a paler color, their coats presenting the greatest differences, for they were of a pale, dirty tint, instead of chestnut brown. The glands on which the cabbage seeds had rested, as well as those bathed by the surrounding secretion, differed greatly in appearance from the other glands on the same leaf, for they all contained brownish granular matter, proving that they had absorbed matter from the seeds. That the secretion acts on the seeds was also shown by some of them being killed, or by the seedlings being injured. 14 cabbage seeds were left for 3 days on leaves, and excited page 387 much secretion. They were then placed on damp sand under conditions known to be favorable for germination. 3 never germinated, and this was a far larger proportion of deaths than occurred with seeds of the same lot, which had not been subjected to the secretion, but were otherwise treated in the same manner. Of the 11 seedlings raised, 3 had the edges of their cotyledons slightly browned, as if scorched, and the cotyledons of 1 grew into a curious indented shape. 2 mustard seeds germinated, but their cotyledons were marked with brown patches, and their radicles deformed. Of 2 radish seeds, neither germinated, whereas of many seeds of the same lot, not subjected to the secretion, all excepting 1 germinated. Of the 2 rumex seeds, 1 died, and the other germinated, but its radical was brown, and soon withered. Both seeds of the avena germinated, 1 grew, while the other had its radical brown and withered. Of 6 seeds of the erica, none germinated, and when cut open, after having been left for 5 months on damp sand, 1 alone seemed alive. 22 seeds of various kinds were found adhering to the leaves of plants growing in a state of nature, and of these, though kept for 5 months on damp sand, none germinated, some being then evidently dead. The effects of objects not containing soluble nitrogenous matter. From the foregoing experiments and observations, we page 389 see that objects not containing soluble matter have little or no power of exciting the glands to secrete non-nitrogenous fluids. If dense, cause the glands to pour forth a large supply of viscid fluid. But this is not in the least acid. On the other hand, the secretion from glands excited by contact with nitrogenous solids or liquids is invariably acid, and is so copious that it often runs down the leaves and collects within the naturally incurved margins. The secretion in this state has the power of quickly dissolving, that is, of digesting the muscles of insects, meat, cartilage, albumin, fibrin, gelatin, and casein, as it exists in the curds of milk. The glands are strongly excited by chemically prepared casein and gluten, but these substances, the latter not having been soaked in weak hydrochloric acid, are only partially dissolved, as was likewise the case with Drosera. The secretion, when containing animal matter in solution, whether derived from solids or from liquids such as an infusion of raw meat milk or a weak solution of carbonate of ammonia, is quickly absorbed, and the glands, which were before limpid and of a greenish color, become brownish and contain masses of aggregated granular matter. This matter, from its spontaneous movements, no doubt consists of protoplasm. No such effect is produced by the action of non-nitrogenous fluids. After the glands have been excited to secrete freely, they cease for a time to secrete, but begin again in the course of a few days. Glands in contact with pollen, the leaves of other plants, and various kinds of seeds pour forth much acid secretion, and afterwards absorb matter probably of an albuminous nature from them, nor can the benefit thus derived be insignificant, for a considerable page 390 amount of pollen must be blown from the many wind-fertilized carices, grasses Roman 100 growing, where pinguicula lives on to the leaves thickly covered with viscid glands, and forming large rosettes. Even a few grains of pollen on a single gland causes it to secrete copiously, we have also seen how frequently the small leaves of Erica tetralix and of other plants, as well as various kinds of seeds and fruits, especially of carex, adhere to the leaves. One leaf of the pinguicula had caught 10 of the little leaves of the Erica, and 3 leaves on the same plant had each caught a seed. Seeds, subjected to the action of the secretion, are sometimes killed or the seedlings injured. We may therefore conclude that Pinguicula vulgaris, with its small roots, is not only supported to a large extent by the extraordinary number of insects which it habitually captures, but likewise draws some nourishment from the pollen, leaves, and seeds of other plants, which often adhere to its leaves. It is therefore partly a vegetable as well as an animal feeder. Pinguicula grandiflora. This species is so closely allied to the last that it is ranked by Dr. Hooker as a subspecies. It differs chiefly in the larger size of its leaves and in the glandular hairs near the basal part of the midrib being longer, but it likewise differs in constitution. I hear from Mr. Ralphs, who was so kind as to send me plants from Cornwall, that it grows in rather different sites, and Dr. Moore of the Glasnevin Botanic Gardens informs me that it is much more manageable under culture, growing freely and flowering annually, whilst Pinguicula vulgaris has to be renewed every year. Mr. Ralphs found numerous page 391 insects and fragments of insects adhering to almost all the leaves. These consisted chiefly of Diptera, with some Hymenoptera, Homoptera, Coleoptera, and a moth on one leaf. There were 9 dead insects, besides a few still alive. He also observed a few fruits of Carex pulicaris, as well as the seeds of this same Pinguicula, adhering to the leaves. I tried only 2 experiments with this species. Firstly, a fly was placed near the margin of a leaf, and after 16 HRS, this was found well inflected. Secondly, several small flies were placed in a row along one margin of another leaf, and by the next morning, this whole margin was curled inwards, exactly as in the case of Pinguicula vulgaris. Pinguicula lusitanica. This species, of which living specimens were sent me by Mr. Ralphs from Cornwall, is very distinct from the 2 foregoing ones. The leaves are rather smaller, much more transparent, and are marked with purple branching veins. The margins of the leaves are much more involuted, those of the older ones extending over 1/3 of the space between the midrib and the outside. As in the 2 other species, the glandular hairs consist of longer and shorter ones, and have the same structure, but the glands differ in being purple, and in often containing granular matter before they have been excited in the lower part of the leaf. Almost half the space on each side between the midrib and margin is destitute of glands, these being replaced by long, rather stiff multicellular hairs, which intercross over the midrib. These hairs perhaps serve to prevent insects from settling on this part of the leaf, where there are no viscid glands by which they could be caught, but it is hardly probable that they were developed for this purpose. The spiral vessels pro page 392 seeding from the midrib terminate at the extreme margin of the leaf in spiral cells, but these are not so well developed. As in the 2 preceding species, the flower peduncles, sepals, and petals are studded with glandular hairs like those on the leaves. The leaves catch many small insects, which are found chiefly beneath the involuted margins, probably washed there by the rain. The color of the glands, on which insects have long lain, is changed, being either brownish or pale purple, with their contents coarsely granular, so that evidently absorb matter from their prey. Leaves of the Erica tetralix, flowers of Agalium, scales of grasses Roman 100 likewise adhered to some of the leaves. Several of the experiments which were tried on Pinguicula vulgaris were repeated on Pinguicula lusitanica, and these will now be given. 1. A moderately sized and angular bit of albumen was placed on one side of a leaf halfway between the midrib and the naturally involuted margin. In 2 HRS 15 Roman 1000, the glands poured forth much secretion, and this side became more enfolded than the opposite one. The inflection increased, and in 3 HRS 30 Roman 1000 extended up almost to the apex. After 24 HRS, the margin was rolled into a cylinder, the outer surface of which touched the blade of the leaf, and reached to within the 120 of an inch of the midrib. After 48 HRS, it began to unfold, and in 72 HRS was completely unfolded. The cube was rounded, and greatly reduced in size, the remainder being in a semi-liquefied state. From the foregoing observations on Pinguicula lusitanica, we see that the naturally much incurved margins of the leaves are excited to curve still farther inwards by contact with organic and inorganic bodies, that albumin, cabbage seeds, bits of spinach leaves, and fragments of glass cause the glands to secrete more freely. That albumin is dissolved by the secretion and cabbage seeds killed by it, and lastly, that matter is absorbed by the glands from the insects, which are caught in large numbers by the viscid secretion. The glands on the flower peduncles seem to have no such power. This species differs from Pinguicula vulgaris and grandiflora in the margins of the leaves, when excited by organic bodies being inflected to a greater degree, and in the inflection lasting for a longer time, the glands also seem to be more easily excited to increased secretion by bodies not yielding soluble nitrogenous matter. In other respects, as far as my observations serve, all three species agree in their functional powers. Page 395. Chapter Romans 17, Utricularia. Utricularia, neglected structure of the bladder. The uses of the several parts number of imprisoned animals. Manner of capture. The bladders cannot digest animal matter, but absorb the products of its decay. Experiments on the absorption of certain fluids by the quadruped processes. Absorption by the glands. Summary of the observation on absorption. Development of the bladders. Utricularia vulgaris, Utricularia minor, Utricularia clandestina. I was led to investigate the habits and structure of the species of this genus, partly from their belonging to the same natural family as Pinguicula, but more especially by Mr. Holland's statement that water insects are often found imprisoned in the bladders, which he suspects are destined for the plant to feed on. The plants, which I first received as Utricularia vulgaris from the New Forest in Hampshire and from Cornwall, and which I have chiefly worked on, have been determined by Dr. Hooker to be a very rare British species, the Utricularia neglecta of Lame. I subsequently received the true Utricularia vulgaris from Yorkshire, since drawing up the following description from my own observations, and those of my son Francis Darwin, an important memoir by Prof. Cohn. The Quart Mag of the High Wycombe Nat Hist Soch, July 1868, p. 5, Del Pino, Alt Osservaz, Sulla Dicogamia, Roman 1,001,869, p. 16, also quotes Crouan as having found 1858 crustaceans within the bladders of Utricularia vulgaris. I am much indebted to the Rev. H. Roman 1000 Wilkinson of Bisterne for having sent me several fine lots of this species from the New Forest. Mr. Ralphs was also so kind as to send me living plants of the same species from near Penzance in Cornwall. Page 396. On Utricularia vulgaris has appeared, and it has been no small satisfaction to me to find that my account agrees almost completely with that of this distinguished observer. I will publish my description as it stood before reading that by Prof. Cohn, adding occasionally some statements on his authority. Fig. 17, Utricularia neglecta branch, with the divided leaves bearing bladders about twice enlarged. Utricularia neglecta, the general appearance of a branch, about twice enlarged, with the pinnatifid leaves bearing bladders, is represented in the above sketch. Fig. 17. The leaves continually bifurcate, so that a full-grown one terminates in from 20 to 30. Beitridge zur Biologie der Pflanzen Dritts Heft, 1875, page 397. Points. Each point is tipped by a short straight bristle, and slight notches on the sides of the leaves bear similar bristles on both surfaces. There are many small papillae crowned with two hemispherical cells in close contact. The plants float near the surface of the water, and are quite destitute of roots, even during the earliest period of growth. They commonly inhabit, as more than one observer has remarked to me, remarkably foul ditches. The bladders offer the chief point of interest. There are often 2 or 3 on the same divided leaf, generally near the base. Though I have seen a single one growing from the stem, they are supported on short footstalks. When fully grown, they are nearly 110 of an inch to Roman 2000 in length. They are translucent of a green color, and the walls are formed of 2 layers of cells. The exterior cells are polygonal and rather large, but at many of the points where the angles meet, there are smaller rounded cells. these latter support short conical projections surmounted by two hemispherical cells in such close apposition that they appear united, but they often separate a little when immersed in certain fluids. The papillae thus formed are exactly like those on the surfaces of the leaves. Those on the same bladder vary much in size, and there are a few, especially on very young bladders, which have an elliptical instead of a circular outline. The two terminal cells are transparent, but must hold much matter in solution, judging from the quantity coagulated by prolonged immersion in alcohol or ether. I infer that this is the case from a drawing of a seedling given by Dr. Warming in his paper Bidrag, till kunskapen om lentibulariaceae, from the Wetenskabelige Meddelelser, Copenhagen, 1874, nose 37 pp. 3358, page 398. The bladders are filled with water. They generally, but by no means always, contain bubbles of air according to the quantity of the contained water, and air they vary much in thickness, but are always somewhat compressed at an early stage of growth. The flat or ventral surface faces the axis or stem, but the footstalks must have some power of movement, for in plants kept in my greenhouse, the ventral surface was generally turned either straight or obliquely downwards. The Rev. H. Roman 1000 Wilkinson examined. Fig 18 Utricularia, neglecta bladder, much enlarged sea collar, indistinctly seen through the walls. Plants for me in a state of nature, and found this commonly to be the case, but the younger bladders often had their valves turned upwards. The general appearance of a bladder, viewed laterally with the appendages on the near side alone represented, is shown in the accompanying figure. Fig 18. The lower side, where the footstalk arises, is nearly straight, and I have called it the ventral surface. The other or dorsal surface is convex, and terminates in two long prolongations formed of several rows of cells containing chlorophyll, and bearing chiefly on page 399, the outside 6 or 7 long pointed multicellular bristles. These prolongations of the bladder may be conveniently called the antennae, for the whole bladder, see Fig 17, curiously resembles an entomostracan crustacean. The short footstalk representing the tail in Fig 18. The near antenna alone is shown beneath the two antennae. The end of the bladder is slightly truncated, and here is situated the most important part of the whole structure, namely the entrance and valve. On each side of the entrance, from 3 to rarely 7 long multicellular bristles project out. Fig 19 Utricularia, neglecta valve of bladder, greatly enlarged. Wards, but only those 4 in number on the near side, are shown in the drawing. These bristles, together with those borne by the antennae, form a sort of hollow cone surrounding the entrance. The valve slopes into the cavity of the bladder or upwards in Fig 18. It is attached on all sides to the bladder, excepting by its posterior margin or the lower one in Fig 19, which is free, and forms one side of the slit-like orifice leading into the bladder. This margin is sharp, thin, and smooth, and rests on the edge of a rim or collar which dips deeply into the page 400 bladder, as shown in the longitudinal section, Fig 20 of the collar and valve. It is also shown at C in Fig 18. The edge of the valve can thus open only inwards, as both the valve and collar dip into the bladder. A hollow or depression is here formed, at the base of which lies the slit-like orifice. The valve is colorless, highly transparent, flexible, and elastic. It is convex in a transverse direction, but has been drawn Fig 19 in a flattened state, by which its apparent breadth is increased. It is formed. Fig 20 Utricularia, neglecta longitudinal vertical section through the ventral portion of a bladder showing valve, and collar V valve. The whole projection above C forms the collar B bifid processes S ventral surface of bladder. According to cone of 2 layers of small cells, which are continuous with the 2 layers of larger cells forming the walls of the bladder, which it is evidently a prolongation. 2 pairs of transparent pointed bristles, about as long as the valve itself, arise from near the free posterior margin Fig 18, and point obliquely outwards in the direction of the antennae. There are also on the surface of the valve numerous glands, as I will call them, for they have the power of absorption, though I doubt whether they ever secrete. They consist of 3 kinds, which page 401, to a certain extent graduate into one another. Those situated round the anterior margin of the valve upper margin in Fig 19 are very numerous, and crowded together they consist of an oblong head on a long pedicel. The pedicel itself is formed of an elongated cell, surmounted by a short one. The glands towards the free posterior margin are much larger, few in number, and almost spherical, having short footstalks. The head is formed by the confluence of two cells, the lower one answering to the short upper cell of the pedicel of the oblong glands. The glands of the third kind have transversely elongated heads and are seated on very short footstalks, so that they stand parallel and close to the surface of the valve. They may be called the two-armed glands. The cells forming all these glands contain nucleus and are lined by a thin layer of more or less granular protoplasm, the primordial utricle of mole. They are filled with fluid, which must hold much matter in solution, judging from the quantity coagulated. After they have been long immersed in alcohol or ether, the depression in which the valve lies is also lined with innumerable glands, those at the sides having oblong heads and elongated pedicels exactly like the glands on the adjoining parts of the valve. The collar, called the peristome by Cohn, is evidently formed like the valve by an inward projection of the walls of the bladder. The cells composing the outer surface, or that facing the valve, have rather thick walls, are of a brownish color, very numerous, and elongated, the lower ones being divided into two by vertical partitions. The whole presents a complex and elegant appearance. The cells forming the inner surface are continuous with those over the whole inner surface of the bladder. The space be page 402 between the inner and outer surface consists of coarse cellular tissue. Fig 20. The inner side is thickly covered with delicate bifid processes hereafter to be described. The collar is thus made thick, and it is rigid so that it retains the same outline, whether the bladder contains little or much air and water. This is of great importance, as otherwise the thin and flexible valve would be liable to be distorted, and in this case would not act properly. Altogether, the entrance into the bladder, formed by the transparent valve, with its four obliquely projecting bristles, its numerous diversely shaped glands, surrounded by the collar-bearing glands on the inside, and bristles on the outside, together with the bristles borne by the antennae, presents an extraordinarily complex appearance when viewed under the microscope. We will now consider the internal structure of the bladder. The whole inner surface, with the exception of the valve, is seen under a moderately high power to be covered with a serried mass of processes. Fig 21. Each of these consists of four divergent arms, whence their name of quadruped processes. They arise from small angular cells. At the junctions of the angles of the larger cells, which form the interior of the bladder, the middle part of the upper surface of these small cells projects a little, and then contracts into a very short and narrow footstalk, which bears the four arms. Fig 22. Of these two are long, but often of not quite equal length, and project obliquely inwards and towards the posterior end of the bladder. The two others are much shorter and project at a smaller angle, that is, are more nearly horizontal, and are directed towards the anterior end of the bladder. These arms are only moderately sharp. They are composed of page 403 extremely thin transparent membrane, so that they can be bent or doubled in any direction without being broken. They are lined with a delicate layer of protoplasm, as is likewise the short conical projection from which they arise. Each arm generally, but not invariably, contains a faintly brown particle, either rounded or more commonly elongated, which exhibits incessant Brownian movements. These Fig 21. Utricularia neglecta, small portion of inside of bladder, much enlarged, showing quadruped processes. Fig 22. Utricularia neglecta, one of the quadruped processes, greatly enlarged. Tickles slowly change their positions and travel from one end to the other of the arms, but are commonly found near their bases. They are present in the quadrupeds of young bladders, when only about a third of their full size. They do not resemble ordinary nuclei, but I believe that they are nuclei in a modified condition, for when absent I could occasionally just distinguish in their places a delicate halo of matter, including a darker spot. Moreover, the quadrupeds of Utricularia montana contain rather larger and much page 404 more regularly spherical, but otherwise similar particles, which closely resemble the nuclei in the cells forming the walls of the bladders. In the present case, there were sometimes 2, 3, or even more nearly similar particles within a single arm, but as we shall hereafter see, the presence of more than one seemed always to be connected with the absorption of decayed matter. The inner side of the collar, c. the previous Fig 20 is covered with several crowded rows of processes differing in no important respect from the quadrupeds. Except in bearing only 2 arms instead of 4, they are, however, rather narrower and more delicate, I shall call them. The bifids they project into the bladder and are directed towards its posterior end. The quadruped and bifid processes, no doubt, are homologous with the papillae on the outside of the bladder and of the leaves, and we shall see that they are developed from closely similar papillae. The uses of the several parts. After the above long but necessary description of the parts, we will turn to their uses. The bladders have been supposed by some authors to serve as floats, but branches which bore no bladders and others from which they had been removed floated perfectly owing to the air in the intercellular spaces. Bladders containing dead and captured animals usually include bubbles of air, but these cannot have been generated solely by the process of decay, as I have often seen air in young, clean, and empty bladders, and some old bladders with much decaying matter had no bubbles. The real use of the bladders is to capture small aquatic animals, and this they do on a large scale in the first lot of plants, which I received from the New Forest early in July. A large proportion of the fully page 405 grown bladders contained prey in a second lot, received in the beginning of August. Most of the bladders were empty, but plants had been selected, which had grown in unusually pure water in the first lot. My son examined 17 bladders, including prey of some kind, and 8 of these contained entomostracan crustaceans, 3 larvae of insects, 1 being still alive, and 6 remnants of animals so much decayed that their nature could not be distinguished. I picked out 5 bladders, which seemed very full, and found in them 4, 5, 8, and 10 crustaceans, and in the 5th a single much elongated larva. In 5 other bladders selected from containing remains, but not appearing very full, there were 1, 2, 4, 2, and 5 crustaceans. A plant of Utricularia vulgaris, which had been kept in almost pure water, was placed by Cone one evening into water swarming with crustaceans, and by the next morning most of the bladders contained these animals entrapped. And swimming round and round their prisons, they remained alive for several days, but at last perished asphyxiated, as I suppose, by the oxygen in the water having been all consumed. Freshwater worms were also found by Cone in some bladders. In all cases, the bladders with decayed remains swarmed with living algae of many kinds, infusoria, and other low organisms, which evidently lived as intruders. Animals enter the bladders by bending inwards the posterior free edge of the valve, which from being highly elastic shuts again instantly, as the edge is extremely thin and fits closely against the edge of the collar, both projecting into the bladder. See section Fig 20. It would evidently be very difficult for any animal to get out when once imprisoned, and apparently they never do escape. To show how closely the edge, page 406 fits, I may mention that my son found a Daphnia, which had inserted one of its antennae into the slit, and it was thus held fast during a whole day on 3 or 4 occasions. I have seen long narrow larvae, both dead and alive, wedged between the corner of the valve and collar, with half their bodies within the bladder and half out. As I felt much difficulty in understanding how such minute and weak animals as are often captured could force their way into the bladders, I tried many experiments to ascertain how this was effected. The free margin of the valve bends so easily that no resistance is felt when a needle or thin bristle is inserted. A thin human hair fixed to a handle and cut off so as to project barely 14 of an inch, entered with some difficulty. A longer piece yielded, instead of entering on 3 occasions. Minute particles of blue glass, so as to be easily distinguished, were placed on valves whilst under water, and on trying gently to move them with a needle, they disappeared so suddenly that, not seeing what had happened, I thought that I had flirted them off. But on examining the bladders, they were found safely enclosed. The same thing occurred to my son, who placed little cubes of green boxwood about 160 of an inch 423 Roman 2000 on some valves, and thrice, in the act of placing them on, or whilst gently moving them to another spot, the valves suddenly opened, and they were engulfed. He then placed similar bits of wood on other valves, and moved them about for some time, but they did not enter again. Particles of blue glass were placed by me on 3 valves, and extremely minute shavings of lead on 2 other valves, After 1 or 2 HRS, none had entered. But in from 2 to 5 HRS, all 5 were enclosed. One of the particles of glass was a page 407 long splinter, of which one end rested obliquely on the valve, and after a few hours it was found fixed half within the bladder and half projecting out, with the edge of the valve fitting closely all round, except at one angle, where a small open space was left. It was so firmly fixed, like the above-mentioned larve, that the bladder was torn from the branch and shaken, and yet the splinter did not fall out. My son also placed little cubes, about 165 of an inch 391 Roman 2000 of green boxwood, which were just heavy enough to sink in water on 3 valves. These were examined after 19 HRS 30 Roman 1000, and were still lying on the valves, but after 22 HRS 30 Roman 1001 was found enclosed. I may here mention that I found in a bladder on a naturally growing plant a grain of sand, and in another bladder 3 grains. These must have fallen by some accident on the valves, and then entered like the particles of glass. The slow bending of the valve from the weight of particles of glass, and even of boxwood, though largely supported by the water, is, I suppose, analogous to the slow bending of colloid substances. For instance, particles of glass were placed on various points of narrow strips of moistened gelatine, and these yielded and became bent with extreme slowness. It is much more difficult to understand how gently moving a particle from one part of a valve to another causes it suddenly to open. To ascertain whether the valves were endowed with irritability, the surfaces of several were scratched with a needle or brushed with a fine camel hair brush so as to imitate the crawling movement of small crustaceans, but the valve did not open. Some bladders, before being brushed, were left for a time in water at temperatures between 80 and 130 F, 266,544, cent, as judging from a wide page 408 spread analogy, this would have rendered them more sensitive to irritation, or would by itself have excited movement, but no effect was produced. We may therefore conclude that animals enter merely by forcing their way through the slit-like orifice, their heads serving as a wedge. But I am surprised that such small and weak creatures as are often captured, for instance the nauplius of a crustacean and a tardigrade, should be strong enough to act in this manner, seeing that it was difficult to push in one end of a bit of a hair 14 of an inch in length. Nevertheless, it is certain that weak and small creatures do enter, and Mrs. Treat of New Jersey has been more successful than any other observer, and has often witnessed, in the case of Utricularia clandestina, the whole process. She saw a tardigrade slowly walking round a bladder. As if reconnoitering, at last it crawled into the depression where the valve lies, and then easily entered. She also witnessed the entrapment of various minute crustaceans. Cyprus was quite wary, but nevertheless was often caught coming to the entrance of a bladder. It would sometimes pause a moment, and then dash away. At other times it would come close up, and even venture part of the way into the entrance and back out, as if afraid another more heedless would open the door and walk in. But it was no sooner in than it manifested alarm, drew in its feet and antennae, and closed its shell. Larvae, apparently of gnats, when feeding near the entrance, are pretty certain to run their heads into the net, whence there is no retreat. A large larva is sometimes 3 or 4 hours in being swallowed, the process bringing to New York Tribune, reprinted in the Guard Cron 1875 p. 303, page 409. Mind what I have witnessed when a small snake makes a large frog its victim, but as the valve does not appear to be in the least irritable, the slow swallowing process must be the effect of the onward movement of the larva. It is difficult to conjecture what can attract so many creatures, animal and vegetable feeding crustaceans, worms, tardigrades, and various larvae, to enter the bladders. Mrs. Treat says that the larvae just referred to are vegetable feeders, and seem to have a special liking for the long bristles round the valve, but this taste will not account for the entrance of animal-feeding crustaceans. Perhaps small aquatic animals habitually try to enter every small crevice like that between the valve and collar in search of food or protection. It is not probable that the remarkable transparency of the valve is an accidental circumstance, and the spot of light thus formed may serve as a guide. The long bristles round the entrance apparently serve for the same purpose. I believe that this is the case because the bladders of some epiphytic and marsh species of Utricularia, which live embedded either in entangled vegetation or in mud, have no bristles round the entrance, and these, under such conditions, would be of no service as a guide. Nevertheless, with these epiphytic and marsh species, two pairs of bristles project from the surface of the valve, as in the aquatic species, and their use probably is to prevent two large animals from trying to force an entrance into the bladder, thus rupturing orifice. As under favorable circumstances, most of the bladders succeed in securing prey. In one case, as many as 10 crustaceans, the valve is so well fitted to page 410 allow animals to enter, and to prevent their escape, and, as the inside of the bladder presents so singular a structure clothed with innumerable quadruped and bifid processes, it is impossible to doubt that the plant has been specially adapted for securing prey. From the analogy of Pinguicula belonging to the same family, I naturally expected that the bladders would have digested their prey, but this is not the case, and there are no glands fitted for secreting the proper fluid. Nevertheless, in order to test their power of digestion, minute fragments of roast meat, 3 small cubes of albumin, and 3 of cartilage were pushed through the orifice into the bladders of vigorous plants. They were left from 1 day to 3 days and a half within, and the bladders were then cut open, but none of the above substances exhibited the least signs of digestion or dissolution, the angles of the cubes being as sharp as ever. These observations were made subsequently to those on Drosera dionea, Drosophilum, and Pinguicula, so that I was familiar with the appearance of these substances when undergoing the early and final stages of digestion. We may therefore conclude that Utricularia cannot digest the animals which it habitually captures. In most of the bladders, the captured animals are so much decayed that they form a pale brown pulpy mass, with their chitinous coats so tender that they fall to pieces with the greatest ease. The black pigment of the eye spots is preserved better than anything else. Limbs, jaws Roman 100, are often found quite detached, and this, I suppose, is the result of the vain struggles of the later captured animals. I have sometimes felt surprised at the small proportion of imprisoned animals in a fresh state, compared with those utterly decayed. Mrs. Treat states, with respect page 411, to the larve above referred to, that usually in less than 2 days after a large one was captured, the fluid contents of the bladders began to assume a cloudy or muddy appearance, and often became so dense that the outline of the animal was lost to view. This statement raises the suspicion that the bladders secrete some ferment hastening the process of decay. There is no inherent improbability in this supposition, considering that meat soaked for 10 minutes in water mingled with the milky juice of the papaw becomes quite tender, and soon passes, as Brown remarks in his Natural History of Jamaica, into a state of putridity. Whether or not the decay of the imprisoned animals is in any way hastened, it is certain that matter is absorbed from them by the quadruped and bifid processes. The extremely delicate nature of the membrane of which these processes are formed, and the large surface which they expose owing to their number crowded over the whole interior of the bladder, are circumstances all favoring the process of absorption. Many perfectly clean bladders, which had never caught any prey, were opened, and nothing could be distinguished with a no-8 object glass of Hartnack within the delicate structureless protoplasmic lining of the arms, excepting in each a single yellowish particle or modified nucleus. Sometimes 2 or even 3 such particles were present, but in this case traces of decaying matter could generally be detected. On the other hand, in bladders containing either one large or several small decayed animals, the processes presented a widely different appearance. 6 such bladders were carefully examined. One contained an elongated coilodup larva, another a single large entomostracan crustacean, and the others from 2 to 5 smaller ones, all page 412. In a decayed state in these 6 bladders, a large number of the quadruped processes contain transparent, often yellowish, more or less confluent spherical or irregularly shaped masses of matter. Some of the processes, however, contained only fine granular matter, the particles of which were so small that they could not be defined clearly with no 8 of Hartnack. The delicate layer of protoplasm lining their walls was in some cases a little shrunk on 3 occasions. The above small masses of matter were observed and sketched at short intervals of time, and they certainly changed their positions relatively to each other, and to the walls of the arms separate masses sometimes became confluent, and then again divided. A single little mass would send out a projection which after a time separated itself. Hence, there could be no doubt that these masses consisted of protoplasm, bearing in mind that many clean bladders were examined with equal care, and that these presented no such appearance. We may confidently believe that the protoplasm in the above cases had been generated by the absorption of nitrogenous matter from the decaying animals in 2 or 3 other bladders, which at first appeared quite clean. On careful search, a few processes were found with their outsides clogged with a little brown matter, showing that some minute animal had been captured and had decayed, and the arms here included a very few more or less spherical and aggregated masses, the processes in other parts of the bladders being empty and transparent. On the other hand, it must be stated that in 3 bladders containing dead crustaceans, the processes were likewise empty. This fact may be accounted for by the animals not having been sufficiently decayed, or by time enough not having been allowed for the generation of proto-page 413 plasm, or by its subsequent absorption and transference to other parts of the plant. It will hereafter be seen that in 3 or 4 other species of Utricularia, the quadruped processes in contact with decaying animals likewise contained aggregated masses of protoplasm. On the absorption of certain fluids by the quadruped and bifid processes, these experiments were tried to ascertain whether certain fluids, which seemed adapted for the purpose, would produce the same effects on the processes as the absorption of decayed animal matter. Such experiments are, however, troublesome, for it is not sufficient merely to place a branch in the fluid, as the valve shuts so closely that the fluid apparently does not enter soon, if at all. Even when bristles were pushed into the orifices, they were in several cases wrapped so closely round by the thin flexible edge of the valve that the fluid was apparently excluded, so that the experiments tried in this manner are doubtful and not worth giving. The best plan would have been to puncture the bladders, but I did not think of this till too late, excepting in a few cases. In all such trials, however, it cannot be ascertained positively that the bladder, though translucent, does not contain some minute animal in the last stage of decay. Therefore, most of my experiments were made by cutting bladders longitudinally into 2. The quadrupeds were examined with no 8 of Hartnack, then irrigated whilst under the covering glass with a few drops of the fluid under trial, kept in a damp chamber, and re-examined after stated intervals of time with the same power as before. 4 bladders were first tried as a control experiment in the manner just described, in a solution of 1 part of gum arabic to 218 of water, and 2 bladders in a solution of 1 part of sugar to 437 of water, and in neither case was any page 414 change perceptible in the quadrupeds or bifids after 21 HRS. 4 bladders were then treated in the same manner with a solution of 1 part of nitrate of ammonia to 437 of water, and re-examined after 21 HRS. In 2 of these, the quadrupeds now appeared full of very finely granular matter, and their protoplasmic lining, or primordial utricle, was a little shrunk. In the 3rd bladder, the quadrupeds included distinctly visible granules, and the primordial utricle was a little shrunk after only 8 HRS. In the 4th bladder, the primordial utricle in most of the processes was here and there thickened into little irregular yellowish specks, and from the gradations which could be traced in this and other cases, these specks appear to give rise to the larger free granules contained within some of the processes. Other bladders, which as far as could be judged, had never caught any prey, were punctured and left in the same solution for 17 HRS, and their quadrupeds now contained very fine granular matter. A bladder was bisected, examined, and irrigated with a solution of 1 part of carbonate of ammonia to 437 of water after 8 HRS 30 Roman 1000. The quadrupeds contained a good many granules, and the primordial utricle was somewhat shrunk after 23 HRS. The quadrupeds and bifids contained many spheres of hyaline matter, and in one arm 24 such. Spheres of moderate size were counted 2 bisected bladders, which had been previously left for 21 HRS in the solution of gum 1 part to 218 of water without being affected, were irrigated with the solution of carbonate of ammonia, and both had their quadrifids modified in nearly the same manner as just described on after only 9 HRS and the other. After 24 HRS, 2 bladders, which appeared never to have caught any prey, were punctured and placed in the solution. The quadrifids of one were examined after 17 HRS and found slightly opaque. The quadrifids of the other, examined after 45 HRS, had their primordial utricles more or less shrunk, with thickened yellowish specks like those due to the action of nitrate of ammonia. Several uninjured bladders were left in the same solution, as well as a weaker solution of 1 part to 1750 of water, or 1 gr to 4 Rs, and after 2 days the quadrifids were more or less opaque, with their contents finely granular, but whether the solution had entered by the orifice or had been absorbed from the outside, I know not. 2 bisected bladders were irrigated with a solution of 1 part of urea to 218 of water, but when this solution was employed I forgot that it had been kept for some days in a warm room, and had therefore probably generated ammonia. Anyhow, page 415, the quadrifids were affected after 21 HRS, as if a solution of carbonate of ammonia had been used, for the primordial utricle was thickened in specks, which seemed to graduate into separate granules. 3 bisected bladders were also irrigated with a fresh solution of urea of the same strength. Their quadrifids after 21 HRS were much less affected than in the former case. Nevertheless, the primordial utricle in some of the arms was a little shrunk, and in others was divided into 2 almost symmetrical sacs. 3 bisected bladders, after being examined, were irrigated with a putrid and very offensive infusion of raw meat after 23 HRS. The quadrifids and bifids in all 3 specimens abounded with minute hyaline spherical masses, and some of their primordial utricles were a little shrunk. 3 bisected bladders were also irrigated with a fresh infusion of raw meat, and to my surprise the quadrifids in one of them appeared after 23 HRS, finely granular, with their primordial utricles somewhat shrunk and marked with thickened yellowish specks, so that they had been acted on in the same manner as by the putrid infusion or by the salts of ammonia in the second bladder. Some of the quadrifids were similarly acted on, though to a very slight degree, whilst the third bladder was not at all affected. From these experiments, it is clear that the quadrifid and bifid processes have the power of absorbing carbonate and nitrate of ammonia and matter of some kind from a putrid infusion of meat. Salts of ammonia were selected for trial, as they are known to be rapidly generated by the decay of animal matter in the presence of air and water, and would therefore be generated within the bladders containing captured prey. The effect produced on the processes by these salts and by a putrid infusion of raw meat differs from that produced by the decay of the naturally captured animals only in the aggregated masses of protoplasm, being in the latter case of larger size. But it is probable that the fine granules and small hyaline spheres produced by the solutions would coalesce into larger masses with time enough allowed. Page 416, we have seen with Drosera that the first effect of a weak solution of carbonate of ammonia on the cell contents is the production of the finest granules, which afterwards aggregate into larger, more or less rounded masses, and that the granules in the layer of protoplasm, which flows round the walls, ultimately coalesce with these masses. Changes of this nature are, however, far more rapid in Drosera than in Utricularia, since the bladders have no power of digesting albumin cartilage or roast meat. I was surprised that matter was absorbed, at least in one case, from a fresh infusion of raw meat. I was also surprised, from what we shall presently see with respect to the glands round the orifice, that a fresh solution of urea produced only a moderate effect on the quadrifids. As the quadrifids are developed from papillae, which at first closely resemble those on the outside of the bladders, and on the surfaces of the leaves, I may here state that the 2 hemispherical cells with which these latter papillae are crowned, and which in their natural state are perfectly transparent, likewise absorb carbonate and nitrate of ammonia, for after an immersion of 23 HRS in solutions of 1 part of both these salts to 437 of water, their primordial utricles were a little shrunk, and of a pale brown tint, and sometimes finely granular. The same result followed from the immersion of a whole branch for nearly 3 days in a solution of 1 part of the carbonate to 1750 of water. The grains of chlorophyll also in the cells of the leaves on this branch became in many places aggregated into little green masses, which were often connected together by the finest threads. On the absorption of certain fluids by the glands on the valve and collar, the glands round the orifices of bladders, which are still young, or which have been page 417 long, kept in moderately pure water, are colorless, and their primordial utricles are only slightly or hardly at all granular. But in the greater number of plants in a state of natureined, we must remember that they generally grow in very foul water, and, with plants kept in an aquarium in foul water, most of the glands were of a pale brownish tint. Their primordial utricles were more or less shrunk, sometimes ruptured with their contents, often coarsely granular or aggregated into little masses. That this state of the glands is due to their having absorbed matter from the surrounding water, I cannot doubt, for, as we shall immediately see, nearly the same results follow from their immersion for a few hours in various solutions. Nor is it probable that this absorption is useless, seeing that it is almost universal with plants growing in a state of nature, excepting when the water is remarkably pure. The pedicels of the glands, which are situated close to the slit-like orifice, both those on the valve and on the collar, are short, whereas the pedicels of the more distant glands are much elongated and project inwards. The glands are thus well placed, so to be washed by any fluid coming out of the bladder through the orifice. The valve fits so closely, judging from the result of immersing uninjured bladders in various solutions, that it is doubtful whether any putrid fluid habitually passes outwards. But we must remember that a bladder generally captures several animals, and that each time a fresh animal enters a puff of foul water must pass out and bathe the glands. Moreover, I have repeatedly found that by gently pressing bladders which contained air, minute bubbles were driven out through the orifice, and if a bladder is laid on blotting paper and gently pressed, water oozes out. Page 418. In this latter case, as soon as the pressure is relaxed, air is drawn in, and the bladder recovers its proper form. If it is now placed under water and again gently pressed, minute bubbles issue from the orifice and nowhere else, showing that the walls of the bladder have not been ruptured. I mention this because Cohn quotes a statement by Treviranus that air cannot be forced out of a bladder without rupturing it. We may therefore conclude that whenever air is secreted within a bladder already full of water, some water will be slowly driven out through the orifice. Hence, I can hardly doubt that the numerous glands crowded round the orifice are adapted to absorb matter from the putrid water, which will occasionally escape from bladders, including decayed animals. In order to test this conclusion, I experimented with various solutions on the glands. As in the case of the quadrupeds, salts of ammonia were tried, since these are generated by the final decay of animal matter under water. Unfortunately, the glands cannot be carefully examined whilst attached to the bladders in their entire state. Their summits, therefore, including the valve collar and antennae, were sliced off, and the condition of the glands observed. They were then irrigated, whilst beneath a covering glass with the solutions, and after a time re-examined with the same power as before, namely no 8 of Hartnack. The following experiments were thus made. As a control experiment, solutions of 1 part of white sugar and of 1 part of gum to 218 of water were first used to see whether these produced any change in the glands. It was also necessary to observe whether the glands were affected by the summits of the bladders having been cut off. The summits of 4 were thus tried, 1 being examined after 2 HRS 30 Roman 1000, and the other 3 after 23 HRS, but there was no marked change in the glands of any of them. 2 summits bearing quite colorless glands were irrigated with a solution of carbonate of ammonia of the same strength, viz. 1 part to 218 of water, and in 5 Roman 1000, the primordial utricles of most of the glands were somewhat contracted. They were also thickened in specks or patches and had assumed a pale, page 419, brown tint. When looked at again, after 1 HR 30 Roman 1000, most of them presented a somewhat different appearance. A third specimen was treated with a weaker solution of 1 part of the carbonate to 437 of water, and after 1 HR, the glands were pale brown and contained numerous granules. 4 summits were irrigated with a solution of 1 part of nitrate of ammonia to 437 of water. 1 was examined after 15 Roman 1000, and the glands seemed affected after 1 HR 10 Roman 1000. There was a greater change, and the primordial utricles in most of them were somewhat shrunk, and included many granules. In the second specimen, the primordial utricles were considerably shrunk and brownish. After 2 HRS, similar effects were observed in the 2 other specimens, but these were not examined until 21 HRS had elapsed. The nuclei of many of the glands apparently had increased in size. 5 bladders on a branch, which had been kept for a long time in moderately pure water, were cut off and examined, and their glands found very little modified. The remainder of this branch was placed in the solution of the nitrate, and after 21 HRS 2 bladders were examined, and all their glands were brownish, with their primordial utricles somewhat shrunk and finely granular. The summit of another bladder, the glands of which were in a beautifully clear condition, was irrigated with a few drops of a mixed solution of nitrate and phosphate of ammonia, each of 1 part, to 437 of water. After 2 HRS, some few of the glands were brownish. After 8 HRS, almost all the oblong glands were brown, and much more opaque than they were before. Their primordial utricles were somewhat shrunk, and contained a little aggregated granular matter. The spherical glands were still white, but their utricles were broken up into 3 or 4 small hyaline spheres, with an irregularly contracted mass in the middle of the basal part. These smaller spheres changed their forms in the course of a few hours, and some of them disappeared by the next morning. After 23 HRS 30 Roman 1000, they had all disappeared, and the glands were brown. Their utricles now formed a globular shrunken mass in the middle. The utricles of the oblong glands had shrunk very little, but their contents were somewhat aggregated. Lastly, the summit of a bladder, which had been previously irrigated for 21 HRS, with a solution of 1 part of sugar to 218 of water, without being affected, was treated with the above mixed solution, and after 8 HRS 30 Roman 1000, all the glands became brown, with their primordial utricles slightly shrunk. 4 summits were irrigated with a putrid infusion of raw page 420 meat. No change in the glands was observable for some hours, but after 24 HRS, most of them had become brownish and more opaque and granular than they were before. In these specimens, as in those irrigated with the salts of ammonia, the nuclei seemed to have increased both in size and solidity, but they were not measured. 5 summits were also irrigated with a fresh infusion of raw meat. 3 of these were not at all affected in 24 HRS, but the glands of the other 2 had perhaps become more granular. One of the specimens, which was not affected, was then irrigated with the mixed solution of the nitrate and phosphate of ammonia, and after only 25 Roman 1000, the glands contained from 4 or 5 to a dozen granules. After 6 additional hours, their primordial utricles were greatly shrunk. The summit of a bladder was examined, and all the glands found colorless, with their primordial utricles not at all shrunk. Yet many of the oblong glands contained granules just resolvable with no aid of Hartnack. It was then irrigated with a few drops of a solution of 1 part of urea to 218 of water. After 2 HRS 25 Roman 1000, the spherical glands were still colorless, whilst the oblong and 2-armed ones were of a brownish tint, and their primordial utricles much shrunk, some containing distinctly visible granules. After 9 HRS, some of the spherical glands were brownish, and the oblong glands were still more changed, but they contained fewer separate granules. Their nuclei, on the other hand, appeared larger, as if they had absorbed the granules. After 23 HRS, all the glands were brown, their primordial utricles greatly shrunk, and in many cases ruptured. A bladder was now experimented on, which was already somewhat affected by the surrounding water, for the spherical glands, though colorless, had their primordial utricles slightly shrunk, and the oblong glands were brownish, with their utricles much, but irregularly shrunk. The summit was treated with the solution of urea, but was little affected by it in 9 HRS. Nevertheless, after 23 HRS, the spherical glands were brown, with their utricles more shrunk. Several of the other glands were still browner, with their utricles contracted into irregular little masses. Two other summits with their glands colorless, and their utricles not shrunk, were treated with the same solution of urea. After 5 HRS, many of the glands presented a shade of brown, with their utricles slightly shrunk. After 20 HRS 40 Roman 1000, some few of them were quite brown, and contained page 421, irregularly aggregated masses. Others were still colorless, though their utricles were shrunk, but the greater number were not much affected. This was a good instance of how unequally the glands on the same bladder are sometimes affected, as likewise often occurs with plants growing in foul water. Two other summits were treated with a solution which had been kept during several days in a warm room, and their glands were not at all affected when examined after 21 HRS. A weaker solution of 1 part of urea to 437 of water was next tried on 6 summits, all carefully examined. Before being irrigated, the first was re-examined after 8 HRS 30 Roman 1000, and the glands, including the spherical ones, were brown. Many of the oblong glands having their primordial utricles much shrunk, and including granules. The second summit, before being irrigated, had been somewhat affected by the surrounding water, for the spherical glands were not quite uniform in appearance, and a few of the oblong ones were brown, with their utricles shrunk. Of the oblong glands, those which were before colorless became brown in 3 HRS 12 Roman 1000 after irrigation with their utricles slightly shrunk. The spherical glands did not become brown, but their content seemed changed in appearance, and after 23 HRS still more changed and granular. Most of the oblong glands were now dark brown, but their utricles were not greatly shrunk. The 4 other specimens were examined after 3 HRS 30 Roman 1000, after 4 HRS and 9 HRS. A brief account of their condition will be sufficient. The spherical glands were not brown, but some of them were finely granular. Many of the oblong glands were brown, and these, as well as others which still remained colorless, had their utricles more or less shrunk, some of them including small aggregated masses of matter. A summary of the observations on absorption. From the facts now given, there can be no doubt that the variously shaped glands on the valve and round the collar have the power of absorbing matter from weak solutions of certain salts of ammonia and urea, and from a putrid infusion of raw meat. Prof. Cohn believes that they secrete slimy matter, but I was not able to perceive any trace of such action, excepting that after immersion in alcohol, extremely fine lines could sometimes be seen radiating from their page 422 surfaces. The glands are variously affected by absorption. They often become of a brown color. Sometimes they contain very fine granules or moderately sized grains or irregularly aggregated little masses. Sometimes the nuclei appear to have increased in size. The primordial utricles are generally more or less shrunk, and sometimes ruptured. Exactly the same changes may be observed in the glands of plants growing and flourishing in foul water. The spherical glands are generally affected rather differently from the oblong and two-armed ones. The former do not so commonly become brown, and are acted on more slowly. We may therefore infer that they differ somewhat in their natural functions. It is remarkable how unequally the glands on the bladders on the same branch, and even the glands of the same kind on the same bladder, are affected by the foul water in which the plants have grown, and by the solutions which were employed in the former case. I presume that this is due either to little currents bringing matter to some glands, and not to others, or to unknown differences in their constitution. When the glands on the same bladder are differently affected by a solution, we may suspect that some of them had previously absorbed a small amount of matter from the water. However this may be, we have seen that the glands on the same leaf of Drosera are sometimes very unequally affected, more especially when exposed to certain vapors. If glands which have already become brown, with their primordial utricles shrunk, are irrigated with one of the effective solutions, they are not acted on, or only slightly and slowly. If, however, a gland contains merely a few coarse granules, this does not prevent a solution from acting. I have never seen page 423 any appearance making it probable that glands which have been strongly affected by absorbing matter of any kind are capable of recovering their pristine, colorless, and homogeneous condition, and of regaining the power of absorbing. From the nature of the solutions, which were tried, I presume that nitrogen is absorbed by the glands, but the modified brownish more or less shrunk and aggregated contents of the oblong glands were never seen by me or by my son to undergo those spontaneous changes of form. Characteristic of protoplasm, on the other hand, the contents of the larger spherical glands often separated into small hyaline globules or irregularly shaped masses, which changed their forms very slowly and ultimately coalesced, forming a central shrunken mass. Whatever may be the nature of the contents of the several kinds of glands, after they have been acted on by foul water or by one of the nitrogenous solutions, it is probable that the matter thus generated is of service to the plant and is ultimately transferred to other parts. The glands apparently absorb more quickly than do the quadruped and bifid processes, and on the view above maintained, namely, that they absorb matter from putrid water occasionally emitted from the bladders, they ought to act more quickly than the processes, as these latter remain in permanent contact with captured and decaying animals. Finally, the conclusion to which we are led by the foregoing experiments and observations is that the bladders have no power of digesting animal matter, though it appears that the quadrupeds are somewhat affected by a fresh infusion of raw meat. It is certain that the processes within the bladders and the glands outside absorb matter from salts of page 424 ammonia, from a putrid infusion of raw meat, and from urea. The glands apparently are acted on more strongly by a solution of urea and less strongly by an infusion of raw meat than are the processes. The case of urea is particularly interesting because we have seen that it produces no effect on Drosera, the leaves of which are adapted to digest fresh animal matter. But the most important fact of all is that in the present and following species, the quadruped and bifid processes of bladders containing decayed animals generally include little masses of spontaneously moving protoplasm, while such masses are never seen in perfectly clean bladders. Development of the bladders. My son and I spent much time over this subject with small success. Our observations apply to the present species and to Utricularia vulgaris, but were made chiefly on the latter, as the bladders are twice as large as those of Utricularia neglecta. In the early part of autumn, the stems terminate in large buds, which fall off and lie dormant during the winter. At the bottom, the young leaves forming these buds bear bladders in various stages of early development. When the bladders of Utricularia vulgaris are about 1100 inch 254 Roman 2000 in diameter or 1200. In the case of Utricularia neglecta, they are circular in outline with a narrow, almost closed transverse orifice leading into a hollow filled with water, but the bladders are hollow when much under 1100 inch in diameter. The orifices face inwards or towards the axis of the plant. At this early age, the bladders are flattened in the plane in which the orifice lies, and therefore at right angles to that of the mature bladders. They are covered exteriorly with papillae of different sizes, many of which have an elliptical outline. A bundle of vessels formed of page 425 simple elongated cells runs up the short footstalk and divides at the base of the bladder. One branch extends up the middle of the dorsal surface, and the other up the middle of the ventral surface. In full-grown bladders, the ventral bundle divides close beneath the collar, and the two branches run on each side to near where the corners of the valve unite with the collar, but these branches could not be seen in very young bladders. Fig 23, Utricularia vulgaris longitudinal section through a young bladder 1100 of an inch in length, with the orifice too widely open. The accompanying figure, Fig 23, shows a section which happened to be strictly medial through the footstalk and between the nascent antennae of a bladder of Utricularia vulgaris 1100 inch. In diameter, the specimen was soft, and the young valve became separated from the collar to a greater degree than is natural, and is thus represented. We here clearly see that the valve and collar are enfolded prolongations of the walls of the bladder. Even at this early age, glands could be detected on the valve. The state of the quadruped processes will presently be described. The antennae at this period consist of minute cellular projections not shown in the above figure, as they do not lie in the medial plane, which soon bear incipient bristles. In 5 instances, the young antennae were not of quite equal length, and this fact is intelligible if I am right in believing that they represent two divisions of the leaf rising from the end of the bladder. For with the true leaves, whilst very young, the divisions are never as far as I have seen strictly opposite. They, page 426, must therefore be developed one after the other, and so it would be with the 2 and 10. At a much earlier age, when the half-formed bladders are only 1300 inch 846 Roman 2000 in diameter, or a little more, they present a totally different appearance. One is represented on the left side of the accompanying drawing, Fig. 24, the young leaves. Fig. 24, Utricularia vulgaris, young leaf, from a winter bud showing on the left side, a bladder in its earliest stage of development. At this age have broad flattened segments with their future divisions represented by prominences, one of which is shown on the right side now. In a large number of specimens examined by my son, the young bladders appeared as if formed by the oblique folding over of the apex and of one margin with a prominence against the opposite margin. The circular hollow between the enfolded apex and enfolded prominence apparently contracts into the narrow orifice, wherein the valve and collar will be developed, the bladder itself being formed by the confluence of the opposed, page 427, margins of the rest of the leaf. But strong objections may be urged against this view, for we must in this case suppose that the valve and collar are developed asymmetrically from the sides of the apex and prominence. Moreover, the bundles of vascular tissue have to be formed in lines quite irrespective of the original form of the leaf, until gradations can be shown to exist between this, the earliest state, and a young yet perfect bladder. The case must be left doubtful. As the quadruped and bifid processes offer one of the greatest peculiarities in the genus, I carefully observed their development in Utricularia neglecta in bladders about 1100 of an inch in diameter. The inner surface is studded with papillae rising from small cells at the junctions of the larger ones. These papillae consist of a delicate conical protuberance, which narrows into a very short footstalk, surmounted by 2-minute cells. They thus occupy the same relative position and closely resemble, except in being smaller and rather more prominent, the papillae on the outside of the bladders, and on the surfaces of the leaves. The two terminal cells of the papillae first become much elongated in a line parallel to the inner surface of the bladder. Next, each is divided by a longitudinal partition. Soon the two half cells thus formed separate from one another, and we now have 4 cells, or an incipient quadruped process, as there is not space for the 2 new cells to increase in breadth in their original plane. The one slides partly under the other. Their manner of growth now changes, and their outer sides, instead of their apices, continue to grow. The 2 lower cells, which have slid partly beneath the 2 upper ones, form the longer and more upright pair of processes, whilst the 2 upper cells form the shorter, page 428, and more horizontal pair, the 4 together forming a perfect quadruped. A trace of the primary division between the 2 cells on the summits of the papillae can still be seen between the bases of the longer processes. The development of the quadrupeds is very liable to be arrested. I have seen a bladder 150 of an inch in length, including only primordial papillae, and another bladder about half its full size, with the quadrupeds in an early stage of development. As far as I could make out, the bifid processes are developed in the same manner as the quadrupeds, excepting that the 2 primary terminal cells never become divided and only increase in length. The glands on the valve and collar appear at so early an age that I could not trace their development, but we may reasonably suspect that they are developed from papillae like those on the outside of the bladder, but with their terminal cells not divided into 2. The 2 segments forming the pedicels of the glands probably answer to the conical protuberance and short footstalk of the quadruped and bifid processes. I am strengthened in the belief that the glands are developed from papillae like those on the outside of the bladders, from the fact that in Utricularia amethystina, the glands extend along the whole ventral surface of the bladder close to the footstalk. Utricularia vulgaris. Living plants from Yorkshire were sent me by Dr. Hooker. This species differs from the last in the stems, and leaves being thicker or coarser, their divisions form a more acute angle with one another. The notches on the leaves bear 3 or 4 short bristles, instead of 1, and the bladders are twice as large, or about 15 of an inch 5 Roman 2000 in diameter. In all essential respects, the bladders resemble those of Utricularia negucta, but the sides of the peristome are perhaps a little more, page 429, prominent, and always bear, as far as I have seen, 7 or 8 long multicellular bristles. There are 11 long bristles on each antenna, the terminal pair being included. 5 bladders containing prey of some kind were examined. The first included 5 cypris, a large copepod, and a diaptomus, the second 4 cypris, the third a single rather large crustacean, the fourth 6 crustaceans, and the fifth 10. My son examined the quadruped processes in a bladder containing the remains of 2 crustaceans, and found some of them full of spherical or irregularly shaped masses of matter, which were observed to move and to coalesce. These masses therefore consisted of protoplasm. Utricularia minor. Fig. 25, Utricularia minor quadruped process, greatly enlarged. This rare species was sent me in a living state from Cheshire through the kindness of Mr. John Price. The leaves and bladders are much smaller than those of Utricularia negucta. The leaves bear fewer and shorter bristles, and the bladders are more globular. The antennae, instead of projecting in front of the bladders, are curled under the valve, and are armed with 12 or 14 extremely long multicellular bristles, generally arranged in pairs. These, with 7 or 8 long bristles on both sides of the peristome, form a sort of net over the valve, which would tend to prevent all animals, excepting very small ones, entering the bladder. The valve and collar have the same essential structure as in the 2 previous species, but the glands are not quite so numerous. The oblong ones are rather more elongated, whilst the 2-armed ones are rather less elongated. The 4 bristles, which project obliquely from the lower edge of the valve, are short. Their shortness, compared with those on the valves of the foregoing species, is intelligible, if my view is correct, that they serve to prevent 2 large animals forcing an entrance through the valve, thus injuring it, for the valve is already protected to a certain extent by the incurved antennae together with the lateral bristles. The bifid processes are like those in the previous species, but the quadrupeds differ in the 4 arms. Fig. 25, page 430, being directed to the same side, the 2 longer ones being central, and the 2 shorter ones on the outside. The plants were collected in the middle of July, and the contents of 5 bladders, which from their opacity seemed full of prey, were examined. The first contained no less than 24-minute freshwater crustaceans, most of them consisting of empty shells, or including only a few drops of red oily matter. The second contained 20, the third 15, the fourth 10, some of them being rather larger than usual, and the fifth, which seemed stuffed quite full, contained only 7, but 5 of these were of unusually large size. The prey, therefore, judging from these 5 bladders, consists exclusively of freshwater crustaceans, most of which appeared to be distinct species from those found in the bladders of the 2 former species. In one bladder, the quadrupeds, in contact with a decaying mass, contained numerous spheres of granular matter, which slowly changed their forms and positions. Utricularia clandestina. This North American species, which is aquatic like the 3 foregoing ones, has been described by Mrs. Treat of New Jersey, whose excellent observations have already been largely quoted. I have not as yet seen any full description by her of the structure of the bladder, but it appears to be lined with quadruped processes. A vast number of captured animals were found within the bladders, some being crustaceans, but the greater number delicate, elongated larvae, I suppose of Culicidae. On some stems, fully 9 out of every 10 bladders contained these larvae or their remains. The larvae showed signs of life from 24 to 36 hours after being imprisoned, and then perished. Page 431. Chapter Roman 18 Utricularia continued. Utricularia Montana. Description of the bladders on the subterranean rhizome spray captured by the bladders of plants under culture, and in a state of nature absorption by the quadruped processes and glands tubers, serving as reservoirs for water. Various other species of Utricularia polypomphylix genlycia. Different nature of the trap for capturing prey. Diversified methods by which plants are nourished. Fig. 26, Utricularia montana. Rhizome swollen into a tuber. the branches bearing minute bladders of natural size. Utricularia montana. This species inhabits the tropical parts of South America and is said to be epiphytic, but judging from the state of the roots rhizomes of some dried specimens from the herbarium at Kew, it likewise lives in earth, probably in crevices of rocks in English hothouses. It is grown in peaty soil. Lady Dorothy Neville was so kind as to give me a fine plant, and I received another from Dr. Hooker. The leaves are entire instead of being much divided. As in the foregoing aquatic species, they are elongated, about 1/12 in breadth and furnished with a distinct footstalk. The plant produces numerous colorless rhizomes as thin as threads, which bear minute bladders, and occasionally swell into tubers, as will page 432 hereafter be described. These rhizomes appear exactly like roots, but occasionally throw up green shoots. They penetrate the earth, sometimes to the depth of more than 2 inches, but when the plant grows as an epiphyte, they must creep amidst the moss's roots, decayed bark Roman 100, with which the trees of these countries are thickly covered. As the bladders are attached to the rhizomes, they are necessarily subterranean. They are produced in extraordinary numbers. One of my plants, though young, must have borne several hundreds, for a single branch out of an entangled mass had 32, and another branch about 2 inches in length, but with its end and one side branch broken off, had 73 bladders. The bladders are compressed and rounded with the ventral surface, or that between the summit of the long delicate footstalk, and valve extremely short. Fig. 27. They are colorless and almost as transparent as glass, so that they appear smaller than they really are, the largest being under the 120 of an inch 1 Roman 2000 in its longer diameter. They are formed of rather large angular cells, at the junctions of which oblong papillae project, corresponding with those on the surfaces of the bladders of the previous species. Similar papillae abound on the rhizomes and even on the entire leaves, but they are rather broader on the latter vessels, marked with parallel bars, instead of by a spiral line run up the footstalks. And Prof. Oliver has figured a plant of Utricularia jamesoniana, Proc. Lin. Soc. Vol. Roman, 4p. 169, having entire leaves and rhizomes like those of our present species, but the margins of the terminal halves of some of the leaves are converted into bladders. This fact clearly indicates that the bladders on the rhizomes of the present and following species are modified segments of the leaf, and they are thus brought into accordance with the bladders attached to the divided and floating leaves of the aquatic species, page 433. Just enter the bases of the bladders, but they do not bifurcate and extend up the dorsal and ventral surfaces as in the previous species. The antennae are of moderate length and taper to a fine point. They differ conspicuously from those before described in not being armed with bristles. Their bases are so abruptly curved that their tips generally rest one on each side of the middle of the bladder. But Fig. 27, Utricularia montana bladder, about 27 times enlarged. Sometimes near the margin, their curved bases thus form a roof over the cavity in which the valve lies, but there is always left on each side a little circular passage into the cavity, as may be seen in the drawing, as well as a narrow passage between the bases of the 2 antennae, as the bladders are subterranean. Had it not been for the roof, the cavity in which the valve lies would have been liable to be blocked up with earth, page 434, and rubbish, so that the curvature of the antennae is a serviceable character. There are no bristles on the outside of the collar or peristome, as in the foregoing species. The valve is small and steeply inclined, with its free posterior edge abutting against a semicircular, deeply depending collar. It is moderately transparent, and bears 2 pairs of short, stiff bristles in the same position as in the other species. The presence of these 4 bristles, in contrast with the absence of those on the antennae and collar, indicates that they are of functional importance, namely, as I believe, to prevent 2 large animals forcing an entrance through the valve. The many glands of diverse shapes attached to the valve and round the collar in the previous species are here absent, with the exception of about a dozen of the 2-armed or transversely elongated kind, which are seated near the borders of the valve, and are mounted on very short footstalks. These glands are only the 34,000th of an inch, 19 Roman 2,000 in length, though so small they act as absorbents. The collar is thick, stiff, and almost semicircular. It is formed of the same peculiar brownish tissue as in the former species. The bladders are filled with water and sometimes include bubbles of air. They bear internally rather short, thick quadruped processes arranged in approximately concentric rows. The two pairs of arms of which they are formed differ only a little in length and stand in a peculiar position. Fig 28, the two longer ones forming one line and the two shorter ones another. Parallel line. Each arm includes a small spherical mass of brownish matter, which when crushed breaks into angular pieces. I have no doubt that these spheres are nuclei for closely similar ones, page 435, are present in the cells forming the walls of the bladders. Bifid processes, having rather short oval arms, arise in the usual position on the inner side of the collar. These bladders therefore resemble in all essential respects the larger ones of the foregoing species. They differ chiefly in the absence of the numerous glands on the valve, and round the collar a few minute ones of one kind alone being present on the valve. They differ more conspicuously in the absence of the long bristles on the antennae, and on the outside of the collar. The presence of these bristles in the previously mentioned species probably relates to the capture of aquatic animals. Fig 28, Utricularia montana, one of the quadruped processes, much enlarged. It seemed to me an interesting question whether the minute bladders of Utricularia montana served, as in the previous species, to capture animals living in the earth, or in the dense vegetation covering the trees on which this species is epiphytic. For in this case, we should have a new subclass of carnivorous plants, namely subterranean feeders. Many bladders therefore were examined with the following results. 1. A small bladder less than 130 of an inch, 847 Roman 2000 in diameter, contained a minute mass of brown much decayed matter, and in this a tarsus with 4 or 5 joints terminating in a double hook was clearly distinguished. Under the microscope, I suspect that it was a remnant of one of the Thysanura. The quadrupeds in contact with this decayed remnant contained either small masses of translucent yellowish matter, generally more page 436, or less globular, or fine granules in distant parts of the same bladder. The processes were transparent and quite empty, with the exception of their solid nuclei. My son made at short intervals of time sketches of one of the above aggregated masses, and found that they continually and completely changed their forms, sometimes separating from one another and again coalescing. Evidently protoplasm had been generated by the absorption of some element from the decaying animal matter. As it appeared probable that this plant would capture a greater number of animals in its native country than under culture, I obtained permission to remove small portions of the rhizomes from dried specimens in the herbarium at Kew. I did not at first find out that it was advisable to soak the rhizomes for 2 or 3 days, and that it was necessary to open the bladders and spread out their contents on glass, as from their state of decay, and from having been dried and pressed, their nature could not otherwise be well distinguished. Several bladders on a plant which had grown in black earth in New Granada were first examined, and 4 of these included remnants of animals. The first contained a hairy acarus so much decayed that nothing was left except its transparent coat, page 437, also a yellow chitinous head of some animal with an internal fork to which the esophagus was suspended, but I could see no mandibles. Also the double hook of the tarsus of some animal, also an elongated, greatly decayed animal, and lastly, curious flask-shaped organism having the walls formed of rounded cells. Professor Klaus has looked at this latter organism and thinks that it is the shell of a rhizopod, probably one of the Arcelidae. In this bladder, as well as in several others, there were some unicellular algae and 1 multicellular alga, which no doubt had lived as intruders. The second bladder contained an acarus much less decayed than the former one, with its 8 legs preserved, as well as remnants of several other articulate animals. A third bladder contained the end of the abdomen with the 2 hinder limbs of an acarus, as I believe a fourth contained remnants of a distinctly articulated bristly animal, and of several other organisms, as well as much dark brown organic matter. the nature of which could not be made out. Some bladders from a plant which had lived as an epiphyte in Trinidad in the West Indies were next examined, but not so carefully as the others, nor had they been soaked long enough. 4 of them contained much brown translucent granular matter, apparently organic, but with no distinguishable parts. The quadruphids in 2 were brownish with their contents granular, and it was evident that they had absorbed matter. In a 5th bladder, there was a flask-shaped organism like that above mentioned. A 6th contained a very long, much decayed worm-shaped animal. Lastly, a 7th bladder contained an organism, but of what nature could not be distinguished. Only one experiment was tried on the quadruphed processes and glands with reference to their power of absorption. A bladder was punctured and left for 24 HRS in a solution of 1 part of urea to 437 of water, and the quadruphed and bifid processes were found much affected. In some arms there was only a single symmetrical globular mass larger than the proper nucleus and consisting of yellowish matter, generally translucent, but sometimes granular. In others, there were 2 masses of different sizes, one large, and the page 430, 8 others small, and in others there were irregularly shaped globules, so that it appeared as if the limpid contents of the processes, owing to the absorption of matter from the solution, had become aggregated sometimes round the nucleus and sometimes into separate masses, and that these then tended to coalesce. The primordial utricle or protoplasm lining the processes was also thickened here and there into irregular and variously shaped specks of yellowish translucent matter, as occurred in the case of Utricularia neglecta. Under similar treatment, these specks apparently did not change their forms. The minute 2-armed glands on the valve were also affected by the solution, for they now contained several, sometimes as many as 6 or 8, almost spherical masses of translucent matter tinged with yellow, which slowly changed their forms and positions. Such masses were never observed in these glands in their ordinary state. We may therefore infer that they serve for absorption whenever a little water is expelled from a bladder containing animal remains, by the means formerly specified, more especially by the generation of bubbles of air. It will fill the cavity in which the valve lies, and thus the glands will be able to utilize decayed matter which otherwise would have been wasted. Finally, as numerous minute animals are captured by this plant in its native country, and when cultivated, there can be no doubt that the bladders, though so small, are far from being in a rudimentary condition. On the contrary, they are highly efficient traps, nor can there be any doubt that matter is absorbed from the decayed prey by the quadruphed and bifid processes, and that protoplasm is thus generated, what tempts animals of such diverse kinds to enter page 439, the cavity beneath the bode and 10, and then force their way through the little slit-like orifice between the valve and collar into the bladders filled with water, I cannot conjecture. Tubers, these organs, one of which is represented in a previous figure, Fig 26 of the natural size, deserve a few remarks. 20 were found on the rhizomes of a single plant, but they cannot be strictly counted, for besides the 20, there were all possible gradations between a short length of a rhizome just perceptibly swollen, and one so much swollen that it might be doubtfully called a tuber. When well developed, they are oval and symmetrical, more so than appears in the figure. The largest which I saw was 1 inch 25 Roman 2000 in length, and 45 inch 11 Roman 2000 in breadth. They commonly lie near the surface, but some are buried at the depth of 2 inches. The buried ones are dirty white, but those partly exposed to the light become greenish from the development of chlorophyll in their superficial cells. They terminate in a rhizome, but this sometimes decays and drops off. They do not contain any air, and they sink in water. Their surfaces are covered with the usual papillae, the bundle of vessels which runs up each rhizome. As soon as it enters, the tuber separates into 3 distinct bundles, which reunite at the opposite end. A rather thick slice of a tuber is almost as translucent as glass, and is seen to consist of large angular cells full of water, and not containing starch or any other solid matter. Some slices were left in alcohol for several days, but only a few extremely minute granules of matter were precipitated on the walls of the cells, and these were much smaller and fewer than those precipitated on the cell walls of the rhizomes and bladders. We may therefore con page 440 conclude that the tuber do not serve as reservoirs for food, but for water during the dry season to which the plant is probably exposed, the many little bladders filled with water would aid towards the same end. To test the correctness of this view, a small plant growing in light peaty earth in a pot only 412 by 412 inches outside measure was copiously watered, and then kept without a drop of water in the hothouse. Two of the upper tubers were beforehand uncovered and measured, and then loosely covered up again. In a fortnight's time, the earth in the pot appeared extremely dry, but not until the third or fifth day were the leaves in the least affected. They then became slightly reflexed, though still soft and green. This plant, which bore only 10 tubers, would no doubt have resisted the drought for even a longer time had I not previously removed 3 of the tubers and cut off several long rhizomes. When on the third or fifth day the earth in the pot was turned out, it appeared as dry as the dust on a road. All the tubers had their surfaces much wrinkled. Instead of being smooth and tense, they had all shrunk. But I cannot say accurately how much, for as they were at first symmetrically oval, I measured only their length and thickness. But they contracted in a transverse line, much more in one direction than in another, so as to become greatly flattened. One of the 2 tubers which had been measured was now 3/4 of its original length and 2/3 of its original thickness in the direction in which it had been measured, but in another direction only 1/3 of its former thickness. The other tuber was 1/4 shorter, 1/8 less thick in the direction in which it had been measured, and only half as thick in another direction. A slice was cut from one of these shriveled tubers, page 441, and examined. The cells still contained much water and no air, but they were more rounded or less angular than before, and their walls not nearly so straight. It was therefore clear that the cells had contracted. The tubers, as long as they remain alive, have a strong attraction for water. The shriveled one from which a slice had been cut was left in water for 22 HRS 30 Roman 1000, and its surface became as smooth and tense as it originally was. On the other hand, a shriveled tuber, which by some accident had been separated from its rhizome, and which appeared dead, did not swell in the least, though left for several days in water. With many kinds of plants, tubers, bulbs, Roman 100 no doubt, serve in part as reservoirs for water, but I know of no case besides the present one of such organs having been developed solely for this purpose. Prof. Oliver informs me that 2 or 3 species of Utricularia are provided with these appendages, and the group containing them has in consequence received the name of orchidioids. All the other species of Utricularia, as well as of certain closely related genera, are either aquatic or marsh plants. Therefore, on the principle of nearly allied plants, generally having a similar constitution, a never-failing supply of water would probably be of great importance to our present species. We can thus understand the meaning of the development of its tubers, and of their number on the same plant, amounting in one instance to at least 20. Utricularia, Nelumbifolia amethystina, Griffitha, Caerulea, Orbiculata multicalis. As I wish to ascertain whether the bladders on the rhizomes of other species of Utricularia and of the page 442 species of certain closely allied genera had the same essential structure as those of Utricularia montana, and whether they captured prey, I asked Prof. Oliver to send me fragments from the herbarium at Kew. He kindly selected some of the most distinct forms having entire leaves and believed to inhabit marshy ground or water. My son Francis Darwin examined them and has given me the following observations, but it should be borne in mind that it is extremely difficult to make out the structure of such minute and delicate objects after they have been dried and pressed. Utricularia nelumbifolia, Organ Mountains, Brazil. The habitat of this species is remarkable according to its discoverer Mr. Gardner. It is aquatic, but is only to be found growing in the water, which collects in the bottom of the leaves of a large tillandsia that inhabits abundantly an arid rocky part of the mountain at an elevation of about 5,000 feet above the level of the sea. Besides the ordinary method, by seed it propagates itself by runners, which it throws out from the base of the flower stem. This runner is always found directing itself towards the nearest tillandsia, when it inserts its point into the water and gives origin to a new plant, which in its turn sends out another shoot. In this manner, I have seen not less than 6 plants united. The bladders resemble those of Utricularia montana in all essential respects, even to the presence of a few minute two-armed glands on the valve within one bladder. There was the remnant of the abdomen of some larva or crustacean of large size. Prof. Oliver has given Proc. Lin Soch Vol Roman for P169 figures of the bladders of two South American species, namely Utricularia jamesoniana and peltata, but he does not appear to have paid particular attention to these organs. Travels in the interior of Brazil, 183,640, 1p. 527, page 443. Having a brush of long, sharp bristles at the apex, other bladders included fragments of articulate animals, and many of them contained broken pieces of a curious organism, the nature of which was not recognized by anyone to whom it was shown. Utricularia amethystina guiana. This species has small entire leaves and is apparently a marsh plant, but it must grow in places where crustaceans exist, for there were two small species within one of the bladders. The bladders are nearly of the same shape as those of Utricularia montana and are covered outside with the usual papillae, but they differ remarkably in the antennae being reduced to two short points united by a membrane hollowed out in the middle. This membrane is covered with innumerable oblong glands supported on long footstalks, most of which are arranged in two rows converging towards the valve. Some, however, are seated on the margins of the membrane, and the short ventral surface of the bladder between the petiole and valve is thickly covered with glands. Most of the heads had fallen off, and the footstalks alone remained, so that the ventral surface and the orifice, when viewed under a weak power, appeared as if clothed with fine bristles. The valve is narrow and bears a few almost sessile glands. The collar, against which the edge shuts, is yellowish and presents the usual structure from the large number of glands on the ventral surface. And round the orifice, it is probable that this species lives in very foul water, from which it absorbs matter as well as from its captured and decaying prey. Utricularia, Griffithae, Malay, and Borneo. The bladders are transparent, and minute one, which was measured, being only 281,000 of an inch 711 Roman 2000 in diameter. The antennae are of moderate length, and page 444. Project straightforward. They are united for a short space at their bases by a membrane, and they bear a moderate number of bristles or hairs, not simple as heretofore, but surmounted by glands. The bladders also differ remarkably from those of the previous species, as within there are no quadruped, only bifid processes in one bladder. There was a minute aquatic larva, in another the remains of some articulate animal, and in most of them grains of sand. Utricularia caerulea, India. The bladders resemble those of the last species, both in the general character of the antennae and in the processes within, being exclusively bifid. They contained remnants of entomostracan crustaceans. Utricularia orbiculata, India. The orbicular leaves and the stems bearing the bladders apparently float. In water, the bladders do not differ much from those of the two last species. The antennae, which are united for a short distance at their bases, bear on their outer surfaces and summits numerous long multicellular hairs surmounted by glands. The processes within the bladders are quadruped, with the four diverging arms of equal length. The prey which they had captured consisted of entomostracan crustaceans. Utricularia multicalis, Sikkim, India, 7,000 to 11,000 feet. The bladders, attached to rhizomes, are remarkable from the structure of the antennae. These are broad flattened, and of large size. They bear on their margins multicellular hairs surmounted by glands. Their bases are united into a single, rather narrow pedicel, and they thus appear like a great digitate expansion at one end of the bladder. Internally, the quadruped processes have divergent arms of equal length. The bladders contained remnants of articulate animals, page 445. Polypomphylix. This genus, which is confined to Western Australia, is characterized by having a quadripartite calyx. In other respects, as Prof. Oliver remarks, it is quite a utricularia. Polypomphylix multifida. The bladders are attached in whorls round the summits of stiff stalks. The two antennae are represented by a minute membranous fork, the basal part of which forms a sort of hood over the orifice. This hood expands into two wings on each side of the bladder. A third wing or crest, appears to be formed by the extension of the dorsal surface of the petiole, but the structure of these three wings could not be clearly made out owing to the state of the specimens. The inner surface of the hood is lined with long, simple hairs containing aggregated matter like that within the quadruped processes of the previously described species. When in contact with decayed animals, these hairs appear therefore to serve as absorbents. A valve was seen, but its structure could not be determined on the collar round the valve. There are in the place of glands numerous one-celled papillae having very short footstalks. The quadruped processes have divergent arms of equal length. Remains of entomostracan crustaceans were found within the bladders. Polypomphylix tenella, the bladders are smaller than those of the last species, but have the same general structure. They were full of debris, apparently organic, but no remains of articulate animals could be distinguished. Proclin Soch Val Roman 4p 171, page 446. Genlycia. This remarkable genus is technically distinguished from Utricularia, as I hear from Prof. Oliver, by having a five-partite calyx. Species are found in several parts of the world, and are said to be Herbae annui paludaceae. Genlycia ornata Brazil. This species has been described and figured by Dr. Warming, who states that it bears two kinds of leaves, called by him spathulate and utricularis. The latter include cavities, and as these differ much from the bladders of the foregoing species, it will be convenient to speak of them as utricles. The accompanying figure Fig 29 of one of the utricularis leaves about thrice enlarged will illustrate the following description by my son, which agrees in all essential points with that given by Dr. Warming. The utricle B is formed by a slight enlargement of the narrow blade of the leaf, a hollow neck, and no less than 15 times as long as the utricle itself, forms a passage from the transverse slit-like orifice O into the cavity of the utricle. A utricle, which measured 136 of an inch 705 Roman 2000 in its longer diameter, had a neck 1536-10 Roman 2000 in length, and 1100 of an inch 254 Roman 2000 in breadth. On each side of the orifice there is a long spiral arm or tube, the structure of which will be best understood by the following illustration. Take a narrow ribbon and wind it spirally round a thin cylinder so that the edges come into contact along its whole length. Then pinch up the two edges so as to form a little crest, which will of course wind spirally. Bidrag til kunskaben om lentibulariaceae, Copenhagen, 1874, page 447. Round the cylinder like a thread, round a screw. If the cylinder is now removed, we shall have a tube like one of the spiral arms. The two projecting edges are not actually united, and a needle can be pushed in easily between them. They are indeed in many places a little separated, forming narrow entrances into the tube. But this may be the result of the drying of the specimens. The lamina of which the tube is formed seems to be a lateral prolongation of the lip of the orifice, and the spiral line between the two projecting edges is continuous with the corner of the orifice. If a fine bristle is pushed down one of the arms, it passes into the top of the hollow neck. Whether the arms are open or closed at their extremities could not be determined, as all the specimens were broken, nor does it appear that Dr. Warming ascertained this point. Fig 29, Genlycia ornata utricularis leaf, enlarged about 3 times L, upper part of lamina of leaf, B utricle, or bladder, N neck, of utricle O orifice, a spirally wound arms with their ends broken off. So much for the external structure. Internally, the lower part of the utricle is covered with spherical papillae formed of 4 cells, sometimes 8, according to Dr. Warming, which evidently answer to the quadruped processes within the bladders of Utricularia, page 448. These papillae extend a little way up the dorsal and ventral surfaces of the utricle, and a few, according to Warming, may be found in the upper part. This upper region is covered by many transverse rows, one above the other, of short, closely approximate hairs, pointing downwards. These hairs have broad bases, and their tips are formed by a separate cell. They are absent in the lower part of the utricle, where the papillae abound. Fig 30, Genlycia ornata, portion of inside of neck, leading into the utricle greatly enlarged, showing the downward pointed bristles and small quadruped cells or processes. The neck is likewise lined throughout its whole length, with transverse rows of long, thin, transparent hairs having broad bulbous fig 30 bases. With similarly constructed sharp points, they arise from little projecting ridges formed of rectangular epidermic cells. The hairs vary a little in length, but their points generally extend down to the row next below, so that if the neck is split open and laid flat, the inner surface resembles a paper of pinched hairs representing the pins, and the little transverse ridges representing the folds of paper through which the pins are thrust. These rows of hairs are indicated in the previous figure 29 by numerous transverse lines crossing the neck. The inside of the neck is page 449, also studded with papillae. Those in the lower part are spherical and formed of 4 cells, as in the lower part of the utricle. Those in the upper part are formed of 2 cells, which are much elongated downwards beneath their points of attachment. These 2-celled papillae apparently correspond with the bifid process in the upper part of the bladders of utricularia. The narrow transverse orifice, O fig 29, is situated between the bases of the 2 spiral arms. No valve could be detected here, nor was any such structure seen by Dr. Warming. The lips of the orifice are armed with many short, thick, sharply pointed, somewhat incurved hairs or teeth. The 2 projecting edges of the spirally wound lamina forming the arms are provided with short incurved hairs or teeth, exactly like those on the lips. These project inwards at right angles to the spiral line of junction between the 2 edges. The inner surface of the lamina supports 2-celled elongated papillae, resembling those in the upper part of the neck, but differing slightly from them, according to Warming, in their footstalks being formed by prolongations of large epidermic cells, whereas the papillae within the neck rest on small cells sunk amidst the larger ones. These spiral arms form a conspicuous difference between the present genus and utricularia. Lastly, there is a bundle of spiral vessels which, running up the lower part of the linear leaf, divides close beneath the utricle. One branch extends up the dorsal, and the other up the ventral side of both the utricle and neck. Of these 2 branches, one enters one spiral arm, and the other branch the other arm. The utricles contained much debris or dirty matter, which seemed organic, though no distinct organisms. Page 450 could be recognized. It is indeed scarcely possible that any object could enter the small orifice and pass down the long narrow neck, except a living creature within the necks, however, of some specimens, a worm with retracted horny jaws, the abdomen of some articulate animal, and specks of dirt, probably the remnants of other minute creatures, were found. Many of the papillae within both the utricles and necks were discolored, as if they had absorbed matter. From this description, it is sufficiently obvious how Genlycia secures its prey, small animals entering the narrow orifice, but what induces them to enter is not known any more than in the case of utricularia would find their egress rendered difficult by the sharp incurved hairs on the lips, and as soon as they passed some way down the neck, it would be scarcely possible for them to return, owing to the many transverse rows of long, straight, downward pointing hairs together with the ridges from which these project. Such creatures would therefore perish either within the neck or utricle, and the quadruped and bifid papillae would absorb matter from their decayed remains. The transverse rows of hairs are so numerous that they seem superfluous merely for the sake of preventing the escape of prey, and as they are thin and delicate, they probably serve as additional absorbents in the same manner as the flexible bristles on the enfolded margins of the leaves of Aldrovanda. The spiral arms no doubt act as accessory traps. Until fresh leaves are examined, it cannot be told whether the line of junction of the spirally wound lamina is a little open along its whole course, or only in parts, but a small creature which forced its way into the tube at any point would be prevented from escaping by the incurved hairs and would find an open path down page 451 the tube into the neck, and so into the utricle. If the creature perished within the spiral arms, its decaying remains would be absorbed and utilized by the bifid papillae. We thus see that animals are captured by Genlycia not by means of an elastic valve, as with the foregoing species, but by a contrivance resembling an eel trap, though more complex. Genlycia africana, South Africa. Fragments of the utriculiferous leaves of this species exhibited the same structure as those of Genlycia ornata. A nearly perfect acarus was found within the utricle or neck of one leaf, but in which of the two was not recorded. Genlycia aurea Brazile fragment of the neck of a utricle was lined with transverse rows of hairs and was furnished with elongated papillae exactly like those within the neck of Genlycia ornata. It is probable, therefore, that the whole utricle is similarly constructed. Genlycia filiformis Bahia, Brazil. Many leaves were examined, and none were found provided with utricles, whereas such leaves were found without difficulty in the three previous species. On the other hand, the rhizomes bear bladders resembling in essential character those on the rhizomes of Utricularia. These bladders are transparent and very small, viz. Only 1100 of an inch, 254 Roman 2000 in length. The antennae are not united at their bases and apparently bear some long hairs on the outside of the bladders. There are only a few papillae and internally very few quadrifid processes. These latter, however, are of unusually large size relatively to the bladder. With the four divergent arms of equal length, no prey could be seen within these minute bladders, as the rhizomes of this species were furnished with bladders. Those of Genlycia africana, ornata, and aurea were carefully page 452 examined, but none could be found. What are we to infer from these facts? Did the three species just named, like their close allies, the several species of Utricularia, aboriginally possess bladders on their rhizomes, which they afterwards lost, acquiring in their place utriculiferous leaves? In support of this view, it may be urged that the bladders of Genlycia filiformis appear from their small size and from the fewness of their quadrifid processes to be tending towards abortion. But why has not this species acquired utriculiferous leaves like its congeners? Conclusion. It has now been shown that many species of Utricularia and of two closely allied genera inhabiting the most distant parts of the world, Europe, Africa, India, the Malay Archipelago, Australia, North and South America, are admirably adapted for capturing by two methods small aquatic or terrestrial animals, and that they absorb the products of their decay. Ordinary plants of the higher classes procure the requisite inorganic elements from the soil by means of their roots, and absorb carbonic acid from the atmosphere by means of their leaves and stems. But we have seen in a previous part of this work that there is a class of plants which digest and afterwards absorb animal matter, namely all the Droseraceae pinguicula, and as discovered by Dr. Hooker Nepenthes, and to this class other species will almost certainly soon be added. These plants can dissolve matter out of certain vegetable substances, such as pollen seeds and bits of leaves. No doubt their glands likewise absorb the salts of ammonia brought to them by the rain. It has also been shown that some other plants can absorb ammonia by page 453, their glandular hairs, and these will profit by that brought to them by the rain. There is a second class of plants which, as we have just seen, cannot digest, but absorb the products of the decay of the animals which they capture, namely Utricularia and its close allies. And from the excellent observations of Dr. Mellichamp and Dr. Canby, there can scarcely be a doubt that Sarracenia and Darlingtonia may be added to this class, though the fact can hardly be considered as yet fully proved. There is a third class of plants which feed, as is now generally admitted, on the products of the decay of vegetable matter, such as the birdsenist Orchis neodia Roman 100. Lastly, there is the well-known fourth class of parasites, such as the mistletoe, which are nourished by the juices of living plants. Most, however, of the plants belonging to these four classes obtain part of their carbon, like ordinary species, from the atmosphere. Such are the diversified means, as far as at present known, by which higher plants gain their subsistence.